I'd like to welcome everyone to our second day of our NCI-sponsored workshop, Synthetic and Systems Approaches to Integrate Spatiotemporal Processes in Cancer. I'm Lucas Bugay. I am a co-chair for today's workshop, together with co-chair Ajit Johnson Nirmal. I will also mention that this is day 2 of a 3-day workshop. This one is focused on tumor-immune interactions. Overall, this is an entire workshop series is co-organized also by Zev Gardner and Laura Heiser, who co-chaired the Tuesday's workshop, which was on tumor initiation. And then next week on Tuesday, we have our third workshop, which is focused on drug resistance. So I'm going to start today with a couple brief overview slides on really the goals and ideas behind this workshop. First, before I start, I should also say this is also co-organized by the NCI. I would say even primarily organized by the NCI, with a large team in particular by Hannah Dueck and Michelle Bernie-Lang, who are really driving this forward. So thank you to them as well. All right, to start today's workshop, a couple intro slides just to define our terms. What are we talking about when we say systems and synthetic biology? Systems biology is, we're defining as a set of approaches and sort of a mindset almost that it integrates experimental and computational generates high-dimensional data to study complex systems, including we're focused here on cancer, and to really create models of those systems that help us better understand them. And when I say models, I mean both quantitative models, but also conceptual models to help us really wrap our heads around these systems. And in synthetic biology, we mean really coming from the other side, more bottom-up, using engineering principles to design and also build biological systems. And whereas systems biology you can think of as really well-suited to generate hypotheses from complex data, synthetic biology is better suited to actually test those hypotheses that arise. And so why are we hosting this workshop on cancer biology and spatial temporal aspects of cancer using systems and synthetic biology? The reason is that across all fields, from cancer biology to systems and synthetic biology, there's been, especially over the past couple of years, this incredible maturation, right? So in cancer biology, we're applying techniques now that we can look at spatial transcriptomics and get the transcriptomes of individual cells in cancer tissues and see really the rich spatial organization, and of course the sort of infer the temporal dynamics that are happening within those systems. Of course, we need the systems biologists to come help us actually formulate models for what actually might be happening there, so we can better understand and address those systems. And at the same time, we're also able to bring in other technologies like multiplex tissue imaging, add in the dynamic component by looking at intravital imaging, looking at things changing over time in actual in vivo. We have other technologies coming from genetic engineering and cell engineering, like lineage tracing, cell recording, and then we have other perturbative technologies more from the field of synthetic biology, things like optogenetics, engineering cells to themselves sense and perturb systems. So there are there's an increasing appreciation of the complexity involved, but also at the same time an increasing sophistication in the tools to really understand that complexity and perturb it and test hypotheses within it. And this is really an area that's been identified as a place where if we bring together these various communities, we can actually hope to make advances faster and that helps better understand complex cancer environments and how to best address them. So that's the theory behind this workshop, and we've identified 3 main themes for this workshop. The first one was tumor initiation or transformation. This is what we covered on Tuesday. A really fantastic day of talks and ideas that emerged. Today, we're focusing on the tumor microenvironment or tumor-immune interactions. And again, we invite everyone to come back on and tell all your friends to come on Tuesday, May 20th, and the focus there will be on therapeutic resistance. What we want everyone to have in mind when we're having these diverse talks in our session today are really thinking about 3 main areas, right? One is, what are the open questions in tumor-immune interactions, particularly in the spatial temporal regulation thereof as pertains to cancer? What opportunities exist to bring in advanced technologies, either from systems biology or synthetic biology, to uniquely address those questions? And if those opportunities don't exist yet, perhaps there are some challenges that stand in the way of implementing them, or maybe there's technologies that are yet to be developed but would be ideal to address some of these questions. What are those? And this is really the substrate for the discussions that we want to bring in throughout the talks and then later in the discussions. We want this to be an interactive workshop. This is a challenge when we have hundreds of people on WebEx. How we are doing this is through a companion platform called Miro, which I did not know of until Tuesday, but it's a really fantastic platform, basically a place for everyone to go and put sticky notes on a shared board. And I believe the link to access Miro is in the chat. So if you go to the link, and we welcome everyone to actually do that now, and go in and just introduce yourself, place your name on one of the sticky notes in the welcome space, name and affiliation and the types of things that you think about or you work on. And then throughout the sessions, we also invite you to write down your ideas as they come to you in a particular setup. So here you see this, the organization is as follows. On the left will be a board for our keynote, which will begin shortly. On the left side of that board, are in the sort of peach sticky notes, we're asking for your questions, okay? And questions about the keynote that we can then bring in for a discussion after that keynote, for a question and answer session. Then in the middle, where the middle section is really focused on opportunities. These challenges, opportunities that relate to the theme of our workshop. Do any come to mind based on this particular talk? And then in the final panel on the right, we're asking you to actually put in some references if that pertain to the comments that you've made. We will have a separate, as you can see, separate board for each conversation that we'll have throughout the day, but in a similar structure: questions, challenges and opportunities, and open questions in the middle, and then references on the right. Okay, this we will then also use in our breakout session later to sort of see discussion with the ideas that resonate most with the audience. Okay, so this is a workshop unlike any other that I have participated in, so I want to run through the agenda briefly. Again, first we will start with our keynote in a number of minutes. That will be a traditional talk like you're used to seeing, about 45 minutes of a talk plus or 40 to 45 minutes, 50 minutes total, including question and answers. Question and answers will be through Miro. So we will not ask you to unmute. We ask you to put your questions in Miro, and the moderator will actually pull questions and ask those questions to the speaker on your behalf. Okay, then after that, there will be 3 different conversations. What this means, each conversation comprises 3 people. There's a moderator, an expert in one of the fields we've identified, and then 2 speakers, 1 speaker from systems biology, 1 speaker from synthetic biology, matched roughly sort of in an interesting way to ask some questions and then give some potential answers. But again, not people that are working together. Really, the idea was to bring in together people who are in different spheres and force them to sort of share ideas and interact in an interesting way. So there will not be a question and answer session after those talks. The moderator will really moderate that discussion. After 3 of those, we will then have a breakout session. Everyone is invited to these breakout sessions. There will be 3 breakout rooms, 1 around each conversation. So you can choose whichever one you find most engaging, go there, and there for the first 10 minutes, you will have an opportunity to interact with everyone, with the speakers, with the moderator, ask questions in this normal way, unmute yourself and have a discussion. And then also after that, we will further discuss these ideas that were populated on the Miro board and try to sort of crystallize or synthesize some of the more interesting ideas. After that, we'll all come back together in a final discussion with a subset of the speakers and moderators and to really discuss some of these larger ideas that have emerged throughout the day. So that's what's in store. I hope it's, you'll find it engaging. If it's anything like it was Tuesday, we're in for a treat. So at the moment, I would like to introduce Dr. Ann Geiger, who is she is the director of the NCI Center for Strategic Scientific Initiatives. to say a few words. And just a word about the center. This really is one of its goals is to support opportunities to connect researchers across disciplines to really identify new opportunities, particularly collaborative ones in cancer research, which is exactly what this workshop is meant to foster. So I turn it over to Dr. Geiger.
Let me say welcome to the second of the 3 days of the workshop. I want to make 3 brief comments and then let the science proceed. The first one is that as NCI staff, as we do during all presidential transitions, we are moving as quickly as possible to implement new directives. And I just want you all to know we continue to advocate for the cancer research enterprise and the scientific community. The second thing I want to do is reiterate that our center is about supporting NCI-wide initiative development, part of which is convening scientists to look for synergies across focus areas, such as synthetic and systems biology. And we do this in collaboration with colleagues. So I want to thank Michelle Burney-Lang from our center, Hannah Dueck from the Division of Cancer Biology for their efforts to coordinate this workshop, as well as a number of other colleagues. I also want to appreciate our center staff, Sean Hanlon, Kelly Crotty, and Andy Kurtz, who you will encounter as facilitators of the breakout discussions, and again, many other NCI colleagues as well. I want to also call your attention to an innovation lab that our center is supporting and that is being organized by the very same team as this workshop. And you can find details about that innovation lab on the slide that is displayed during the breaks. And my third comment is a little selfish, but my background is in population science and public health. But my college concentration was biology. And so Tuesday, I really enjoyed getting back to those roots. And I hope all of you are as excited as I am about today and the third session next week, and ultimately the potential contributions of this research to reducing cancer morbidity and mortality. So with that said, please proceed with the workshop.
Thank you so much, Anne. Hi, everyone. My name is Ajit Johnson Nirmal. Good morning, good afternoon, or good evening, wherever you're in the world right now. And it's my honor to kick off today's scientific presentation. Our keynote speaker is Thea Tilsty. I'm not sure if she is highlighted yet, but I'll quickly introduce her. It's my real honor to introduce Dr. Thea Tilsty. She's a professor of pathology at UCSF and director of Center of Translational Research in Molecular Genetics of Cancer. Dr. Tilsty is internationally recognized for her pioneering work in cancer biology, particularly in elucidating the roles of genetic instability and stromal epithelial interactions in tumorigenesis. And her research has led to the development of clinically adopted diagnostic tests for DCIS, which is actually now worldwide used. And her scientific contributions have earned her numerous honors, including NCI's Outstanding Investigator Award, the Nakahara Award, recognitions such as Komen Scholar, A1 Scholar, and more recently, she was also the recipient of Cancer Research UK Grand Challenge Award. So please join me in welcoming Dr. Tilsty. Thea, it's a real pleasure to have you here, and over to you.
And just quickly, Thea, you may need to click unmute. It trickily may have lost your audio for a minute.
Share again. Sorry about that. Share. Wow. Yes. And may I ask, can you see my slides?
Yes, and they're lovely.
Okay. And it's in presenter mode, yes?
Yes, it is.
Wonderful. Thank you. And thank you for the opportunity to present at this fabulous meeting I attended Tuesday. And I must say that the bar was set very high, and I hope to do it justice. The talk that I'm going to give today could easily have been the first day, this day, or the third workshop. I'm going to talk about insights provided by microenvironmental interactions, specifically how one can reprogram stromal signals to actually control malignancy. And the two pictures on this first slide are two extremely talented young investigators that I had the good fortune to work with. So I am trying to go forward with this, and here. So currently, our view of cancer is epithelial-centric. This is a quote from Sir Michael Stratton. All cancers originate from a single cell that starts to behave abnormally due to the acquired somatic mutations in its genome. I could find the same quote in 2025. This has been codified as the somatic mutation theory, and the idea is through an accumulation of mutations, we eventually arise at a malignant state. This is very important because it's informed the gestalt of the whole field. Here I'm talking about precision medicine. It's taken us to current therapy. The hypothesis is if we can actually identify how to mediate these mutations, we can actually help therapy and control cancer. One of the problems with this is that so far it hasn't quite met its promise. Success rate is about 3 to 8%. This type of an approach also informs some of the immunotherapy that's taking place. Currently, only a subset of patients receiving treatment respond, and some that do respond often relapse after an initial response. There's a lot of issues with resistance and toxicity, but please note that each of these 4 approaches to immunotherapy, they all center in the middle to an immunosuppressive tumor microenvironment. And that's what I'm going to talk about today. How does that come about? So, as I said before, it's epithelial centric. Despite all of the focus on the genomic epithelial changes, we haven't made enough progress. Why? I would say that data has been accumulating for several decades that is inconsistent with a somatic mutation theory. I'm going to give you a couple of examples, and that perhaps we should look a little bit more deeply. And so here I wanted to show you that actually for decades, 3 decades minimally, we have known that there is normal tissue, phenotypically normal tissue that carries cancer mutations. This was first described by Helene Smith in the 90s. Doug Brash did the same with skin. She did it with breast. Most recently, this has been popularized by this study done in the human esophagus, where the picture that I'm showing you here on the left is early, in the middle is middle-aged, and on the right are older-aged people. And the size of the bubble indicates the size, the clonal expansion. The color of the bubble indicates which oncogene mutation this is carrying, this clone is carrying. And I have to emphasize again, all of these look phenotypically normal. So this says epithelial mutations alone are not sufficient to cause cancer. But here is an example from viral carcinogenesis. By this time, 1990, we knew that the Rous sarcoma virus could easily form cancers, but here's a chick that's expressing Rous sarcoma virus in all the cells of its body, yet there are no sarcomas. But if this chick gets wounded, scratched in any way, if it has inflammation, now at that particular site, there's 100% incidence of sarcomas, indicating that the oncogenic viruses by themselves are not sufficient. You need something else. And I'll finish with this third example, more recently, 2007, where by this time we were able to conditionally express oncogenes in certain tissues. And here in this Barbacid study, they expressed activated KRAS mutation in adult pancreatic cells, the acinar cells, but there was no pancreatic cancer until the animals were provoked to get chronic pancreatitis. At that point, 100% of them developed pancreatic cancer. Again, oncogenes alone are not sufficient. The bottom says it's a combination of genetic and non-genetic events that seem to be really important. So if one looks at the tissue, and on the left, I'm showing normal human breast tissue. On the right, I'm showing a human breast cancer. Of course, you can see that the epithelial cells have changed quite dramatically from their proper ductal architecture. But if you look at the stroma, you see that that is just as prominently disrupted as well, both in terms of architecture and composition. And so this leads us to look at the stroma. The epithelial cells have gained a lot of attention, but look at what's happening underneath as the tissue progresses from normal all the way to cancer. You can see that the fibroblasts change, the immune cells change, which is, we'll talk about that today. The ECM components change, the vascular changes. So there's a lot of changes that are happening in the stroma. Many people think that it's reactive. That's the word that's used. But in actuality, it actually occurs much, much before you even see a tumor. And so it's been our contention that all three of these, the epithelium, the ECM, and the stromal cells, are important in order to attain a malignant phenotype. We started to study this in the last century when we asked, what is the contribution of fibroblasts to initiation or progression of cancer? And here what we were able to do is to take human tissues, normal human prostatic cells, epithelial cells, as well as fibroblasts. We immortalized these epithelial cells in a variety of ways. It didn't make a difference for the end result. They're immortalized, but they're non-tumorigenic. And we isolated the fibroblasts from a prostatic adenocarcinoma. We called these carcinoma-associated fibroblasts. Each of these four cell populations alone are not tumorigenic. And so what we did is we combined them in a pairwise fashion in a little bit of Matrigel, slipped it under the renal capsule of a mouse, and asked, is there any effect of the fibroblasts on the epithelial cells? And the results were actually quite striking. If you combine the normal epithelials with the normal fibroblasts, what you do is recapitulate a tiny little piece of human tissue in the renal capsule of the mouse. This structure secretes PSA. When we did these experiments with breast and allowed the mice to get pregnant, these cells even secreted human milk. So normal allows you to recapitulate human tissue. If we put the normal fibroblasts interacting with the initiated epithelial cells, you had an aborted ductal structure. Nothing grew. If we took these carcinoma-associated fibroblasts and allowed them to converse with the normal prostatic epithelial cells, we did get a ductal structure, but it was not normal. It looked exactly like PIN, prostatic intraepithelial neoplasia, or a precancer. And when we did this with breast, we got DCIS looking lesions. So that said that these fibroblasts were sending signals that were picked up by the epithelial cells that changed their phenotype. When we put the fibroblasts, the carcinoma-associated fibroblasts with initiated cells, we got a structure that was 500 times larger than the other structures that I've described. And the signals that were being sent told those cells to proliferate, to trigger angiogenesis and mutagenesis, turn off apoptosis. And you can see here that the epithelial architecture is completely disrupted. This looks exactly like a prostatic adenocarcinoma. And so for the last several years, what we've been doing is trying to understand what are these signals coming from these carcinoma-associated fibroblasts that actually allow initiation and progression, and are there ways that we can actually inhibit that so that we can have a clinical benefit from this? And what I'm going to tell you today is that, yes, we've answered this question, and I'm going to share that with you. But before I do, I wanted to tell you that other people were looking at the stroma at that time as well. So here's a publication from 2000 where Mary Helen Barcelos-Hoff irradiated a cleared mammary fat pad. And so she has the control cleared pad and the irradiated, damaged. And when she introduced the epithelial cells to the cleared fat pad, the one that had been irradiated grew cancers. The control, not so much, which means that the injured stroma provoked the outgrowth of the cancer. This is a beautiful experiment by Mary Hendricks. What she did, this is now looking at the ECM. And she took aggressive melanoma cells, put them on a plate, allowed them to set down a matrix, and then decellularized the plate, removing all live cells, etc. On top of that matrix, she put normal melanocytes, watched them for several days, and what she found out is that the normal melanocytes now acquired tumorigenic phenotypes. She looked at a majority, many of them. This is the one that's so easy to show you today. This is the tumor cells, the melanoma cells expressing tumor proteins. You can see that the normal melanocyte does not, but when you put the normal melanocyte on a tumor ECM, you now turn on these oncogenic proteins, all in the absence of any genetic mutations, telling you that the malignant phenotype can be controlled by what kind of matrix they're on. So a lot of people have been intensely studying stromal components that facilitate progression, drug resistance, metastasis, etc. But many people forget that the stromal components actually can also inhibit malignancy and in some cases revert it. And the most striking example was seen 50 years ago. I'm going to repeat that, 50 years ago from Beatrice Mintz, where she took tumor cells, she put them into the blastocyst of a mouse and found out that the tumor contributed genetic material to the live pups that were born. The tumor came from an agouti mouse, kind of striped, and the mother was black. And here you see this pup. Of course, they used both molecular as well as phenotypic traits to make this point. This pup did not have cancer. It went on to grow up. It was perfectly fertile. It had family of its own, telling us that the microenvironment can control malignant phenotypes. This was also shown by Mina Bissell, where here she's taking non-tumorigenic breast epithelial cells, putting them in matrigel. What they do is they form properly mammospheres, properly controlled for growth, death, polarity, etc. And when she took tumor cells and put them in matrigel, they showed exactly what one would expect, a chaotic, non-regulated group of cells. If she took these cells, which on the right I'm showing you, they have many mutations and karyotypic abnormalities, and now all you do is allow them to interact with an integrin antibody, which changes their relationship to the microenvironment. These cells are now, quote unquote, normalized. Their growth is controlled, their death, their polarity. And if you sequence these, they have all these genetic mutations. If you remove the antibody, the phenotype goes back to the tumor-like malignant phenotypes, back and forth, just dependent on whether you've put in the antibody, telling you that the microenvironment clearly controls the phenotype and overrides the genotype. So, one last example, very quickly, is that, let me go back, is that about 40 years ago, that some investigators took breast cancer cells, they put them on normal stroma, normal fibroblasts. They lost their malignant properties and started expressing proper differentiated functions like ER alpha. This experiment was repeated again by Don Engber's group about 13 years ago, 12 years ago, and he got the same results, but he wanted to take it forward. He wanted to ask, what's coming from the stroma? Is it soluble or insoluble that's allowing this repression of malignant phenotypes? And so here are the tumor cells grown in matrigel. You can see that they're disorganized, quite prolific, etc. If you put on conditioned media from those normal fibroblasts, this is called embryonic murine mesenchyme, that's the EMM, what you see is that there is no normalization. But if he puts these tumor cells on the decellularized ECM of that normal stroma, now you get proper luminal formation. The cells are polarized. They even start expressing ER alpha. So they've been normalized. If you put it on the ECM of a calf, they are not normalized. So that says that there's something special here. Engber's group compared these two ECMs using proteomics, found some differences and commonalities. And I'll give you one example. The biglycan here, if it's expressed by these fibroblasts, you get normalization of the malignant phenotype. If you knock out the biglycan, you ablate that. Not all of these work. Here's tenacin. It does not normalize. So that tells you that there are ECM proteins that can actually control the malignant phenotypes. What he then did is he synthesized biglycan, put it in a syringe, and injected it into the tumor of a mouse. And what happened is the tumor was 1/10 of what it was previously. And when he cut it open and looked at it, the cells had differentiated and were expressing proper breast differentiated proteins, telling you the ECM can control malignant phenotypes. So what I've told you is that epithelial cells are not sufficient to cause cancer by themselves. I've told you that stromal signals can facilitate cancer formation and that they can also repress cancer formation. So today I want to tell you, when do those stromal changes emerge? How do they come about? I want to say that these stromal changes can be prevented and reversed. And then finally, I want to talk about how insights of these stromal changes provide insights into the mechanisms of the earliest biology and tumorigenesis, and gives us insights into how to address some of the challenges with immunotherapy. And I'm going to start at the beginning. Most people start with carcinoma-associated fibroblasts and a fully formed tumor. I'm going to go all the way to the beginning to start the story and continue it. This is difficult because of the many somatic cells in our body. Only a few of them ever find themselves on a path to malignancy. The initiation of cancer is sporadic in time and place, so it's really hard to study. But nature has given us a clue that has given us an edge here. Nature tells us that in injured tissues, they're at very high risk for developing subsequent cancers. Here are some examples of injuries. I'm going to focus on chronic inflammation and ask, how does chronic inflammation increase the risk of cancer, with all these questions that I asked previously. We were fortunate enough to receive this CRUK Grand Challenge Award. I put together a team, an international team, and our job was to understand in mechanistic forms, how does chronic inflammation allow the initiation, progression, increased incidence of cancer, and then use this for clinical benefit. Chronic inflammation is seen in all cancers. As soon as you disrupt tissue architecture, you call the immune cells and you have chronic inflammation. And we know that it's important in progression and metastasis. The cancers that we focused on are actually initiated by chronic inflammation. So for instance, acid reflux. 30% of the Western world suffers from acid reflux, where the stomach and the bile acids come up into the bottom of the esophagus, and that is at increased risk for cancer. What happens with these types of cancers, you can have chronic alcoholism injuring the liver, you can have bacteria injuring the stomach, etc. What happens is, in these areas of chronic injury, the body mounts an adaptive response, a protective response known as metaplasia. There's actually a change in cell identity so that the cells that are there are better able to withstand the injury. Some of these metaplastic lesions go on to dysplasia and to cancer, and these cancers are the worst of all cancers. They're responsible for over 1/4 of cancer deaths worldwide, while these chronic inflammation associated cancers actually only represent 6% of cancers, telling you that these are very lethal. These include pancreatic cancer, high-grade serous ovarian cancer, esophageal adenocarcinoma, etc. So here's an example of a person that has been exposed either to cigarette smoke or air pollution. Here I'm showing you the ciliated and secretory, that's where the secretory cells would be, in the bronchial airway. And here's the cartoon. When this airway is injured, these cells are replaced by squamous cells. They're better able to deal with the injury. And below, I show you the actual histology. You can see this beautiful abrupt transition between the ciliated secretory airway and the squamous airway. The reason that this is so exciting and important is because these types of cancers provide a unique advantage. I've just shown you that when you injure the normal epithelium of the airway, what you have is squamous metaplasia, cells with a new identity. The dysplasia that comes out of this is squamous. And the cancer that comes out of this is squamous lung cancer. 35% of all lung cancers are squamous lung cancers. They come from squamous metaplasia, which tells you that chronic inflammation, this injury, generates this metaplastic lesion directly linked to the increased cancer type. And it thus alerts us to the time and the place that the progression is initiated and then goes forward. It allows us to study the earliest biology. And these lesions can be detected by endoscopy, and they can be collected by biopsy. And there's enough time between the appearance of the metaplasia and the development of the squamous lung cancer that you can actually think about providing some prevention and or intervention in here. So it's a very unique advantage. And what we did with this grant is we took 4 examples. I'm showing you 3 here. Here is the squamous cells of the esophagus. They are replaced by these mucus-secreting cells, the gastrointestinal cells, that then protect the lower esophagus from that acid reflux. Infrequently, they go to dysplasia, and from dysplasia to invasive cancer. Here is the bronchial ciliated secretory cells, going from squamous metaplasia all the way to squamous lung cancer. And then H. pylori bacterial infection in the stomach injures it and allows the stomach cells to be replaced, the gastric to be replaced by intestinal. All of these chronic inflammation associated cancers, not only the 3, but the others that I could mention, have high lethality, very, very low survival, lack effective therapies, and they're often highly metastatic, meaning the majority of these cancers are diagnosed at stage 4. In other words, metastases are throughout the body already. And if you look early enough, they actually share a pattern of genetic and epigenetic alterations. This led us to believe that we're not studying multiple types of cancers, but one disease process. And that if we unlocked the secrets to one, we would unlock the secrets to the others. And that actually turned out to be true, as I'm going to show you. So our hypothesis at a glance is that when you have chronic injury, you're not only injuring the epithelial cells, but you actually have 3 pillars of pathology. You're also changing the stromal signals, how they interact with the epithelial cells, and you're changing quite dramatically the ECM and its characteristics. And so, as I said before, most people study tumor-immune interactions in the context of the fully formed tumor. I'm going to take you right back to the very beginning and show you how that's set up. And I'm going to show you that if you injure these fibroblasts in a variety of ways that I'll discuss, you actually change the fibroblast state. It now enters a state that can cause initiation and progression of the overlying epithelial cells. This state is induced by stress, injury, and what it does is it causes tension instructive signals. So we've called these fibroblasts stiffs. I'm going to show you that it works through mechanotransduction. And what this fibroblast state does is it activates the embryonal programs of epithelial stemness and motility. And it also creates a pro-tumorigenic and immunosuppressive microenvironment right from the very beginning. I'm going to use the airway as the example today, taking normal respiratory bronchial airway, going all the way there. And as I said, we've done this with these other tissue types as well, and getting clinical samples from patients from our collaborator in Montreal, Dr. Lorenzo Ferri. We're able to get biopsies of these phases of going to the disease, and we can do single cell RNA sequencing. So not only can I tell what immune cells are there, or endothelial cells or fibroblasts, I can also tell the epithelial cells, and I can tell what state they're in. That has companion ECM proteomics, so we can see how that is changing as we go from left to right. With Gary Nolan, what we've done is transcriptional spatial transcriptomics with Codex. And much as I showed you for those prostate and breast studies. We can isolate derivatives of these cells and do mix and match experiments and ask what happens. All of this is to generate in vitro and in vivo preclinical models so that we can attain our clinical goals, which is to identify which of these pre-malignant lesions are going to go forward to dysplasia. Can we find a way to prevent this? I'm going to show you that we can do both of those. And then ultimately, swing for the fences, can we actually suggest a therapy for these cancers that don't currently have therapies? What drives malignant progression? So using the single cell sequencing information, you can see that healthy tissue has robust conversations amongst all the cells that are in that tissue. By the time you get to a cancer, notice that the fibroblasts are talking quite animatedly to the immune cells and the cells that have changed cell identity, pointing us to the direction of the fibroblasts. So what Deng Pan did is we obtained the clinical samples from patients. We separate out the epithelial cells and the stromal cells. We combine them in a variety of ways. I'm going to tell you today about air-liquid interface, as well as the recombinants in the renal capsule, as I described before. And the whole idea here was to recapitulate a functional human bronchial airway with ciliated, secretory, basal cells, etc., so that we could identify control points for malignancy progression and obtain mechanistic insights. After several years of work, what Deng was able to do is take the healthy bronchial airways, co-culture them in this air-liquid interface with healthy bronchial fibroblasts, and this is what he generated. He generated a fully differentiated functional human bronchial structure. It even had barrier function. The cilia beat. They all beat in the same direction at the same time and the same rate that it does in the human being. It has all of the proper cells that one would have. And here's a picture of the cilia looking down on this structure on the plate. So having been able to generate a functional and working respiratory epithelium, bronchial epithelium, which we asked, are there ways that we can cause initiation or progression of cancer? We went back to our trick of using calves. And so now what Deng is doing is he's co-culturing these bronchial airway cells from human beings with calves from squamous cell lung carcinoma. And this is what he gets, the induction of squamous metaplasia. The bronchial ciliated and secretory cells are sloughed off, much as you've seen in vivo in the human being, and it's replaced by these squamous cells. I'm going to show you staining of this. Here, the green are the cilia. You can see that they're all but gone in this area of metaplasia. You can see the pink basal cells show basal cell hyperplasia, and now you see the expression of squamous markers. So the differentiation markers for bronchial airway goes down. The differentiation markers for squamous tissue is acquired simply by co-culturing these with calves. Can we get it to go further beyond metaplasia to dysplasia? Here is the co-culture of the normal epithelial cell, bronchial epithelial cells with the normal fibroblasts. Here is the normal epithelial cells with calves. You can see the robust squamous metaplasia. And the next slide, you're going to see it's at the same magnification, added stress, and this fulfills all of the conditions that a pathologist would use to diagnose metaplasia. An increased thickening of the epithelium. You can see the pleomorphy of cell size. Some are very tiny, some are very large, loss of maturation. We even have the generation of chromosomal abnormalities. So we're able to go all the way to dysplasia. This cartoon makes an important point. Here are the bronchial airway cells grown either alone or with the normal fibroblasts. You can see this functional structure that I described to you. When you put them, co-culture them with calves, you see the induction of squamous metaplasia, both in vitro and in vivo. What we then did is we took those epithelial cells and we expressed HPV-16, which ablate P53 and PRB activity, two important tumor suppressors. This first surprise was that these cells looked exactly like these. there was no difference that you could see. They were phenotypically normal, much as I described at the beginning, where you have these cells that are phenotypically normal but carry oncogenic mutations. So we took these cells now and that look normal, and we co-cultured them with calves, these same calves, and now what you got was the induction of metaplasia, both in vitro and in vivo. So we went further, and now what we're doing, aside from that, is looked at other types of injuries. Here are the airway cells co-cultured with the normal fibroblasts. You can see that in the absence of any injury, they're fine. There's no expression of squamous markers. If we now injure this with physical carcinogens, chemical carcinogens, ROS, TGF-beta, we get this robust induction of squamous metaplasia, telling us that chronically injured fibroblasts turn on this state, this stiff state, that then induces metaplasia and dysplasia. I'm just not showing you that. So what's the mechanism? How does this happen? What controls cell identity change? And how do these cells become precancerous? So there's 4 languages that control cell identity. Some of them are through secreted factors. Some of them are non-secreted factors. We tested both. It does not work through secreted factors. It does work through the non-secreted factors. Remember the experiments I showed you about ECM. If you take the normal bronchial epithelial cells and put them on the decellularized ECM of the calf, you now induce metaplasia. And so we took this further and we converged our proteomic data, our secretomic data, all of the single cell sequencing data, and we're comparing the fibro calves with the normal fibroblasts. You'll notice there's a lot of collagen chatter here. And what we did is we focused on SERPIN-H1, otherwise known as HSP47. Why? This is a chaperone that's necessary for collagen maturation. And you can see in these 21 clinical samples, it's pretty much upregulated. This is what it looks like. Here is the normal human bronchial airway. There is no green signal there. The green signal is for HSP47 expression, and here you can see it in a subpopulation of fibroblasts in this squamous lung cancer. And by the way, this would be the same if I showed you pancreatic cancer, esophageal cancer, bladder cancer, anything. There's a subset of fibroblasts that expresses HSP47 in all of those cancers. HSP47, as I said before, allows for the maturation of collagen as well as the secretion and bundling, which tells us that it's very important. And then we had another important hint, and that was the cells that robustly induced metaplasia had a high expression of this protein. The ones that were very weak at doing that had a very low expression. So it sets us up for the perfect genetic experiment of necessity and sufficiency. And so what we did is we took these fibroblasts, knocked down the HSP47, and those fibroblasts no longer induce metaplasia or dysplasia. If we take these fibroblasts that could not induce metaplasia and now overexpress HSP47, they now robustly induce metaplasia and dysplasia, telling us that the expression of HSP47 is sufficient and the expression of HSP47 is necessary in order to induce the initiation and progression of these types of cancers. This is a small molecule inhibitor. Here's the epithelial cells with the normal fibroblasts, with the calves. You can see the induction of the squamous metaplasia. If we put in the inhibitor of HSP47, there is no induction of squamous metaplasia. And even more interesting, if we take this structure, which takes about 2.5 to 3 weeks to form, and once it's fully formed, we put on the COL-003, the inhibitor, it now reverts to this ciliated secretory bronchial airway that was originally seen. So that means that HSP47 inhibitors can not only prevent the movement, the induction of metaplasia, it can also revert it back to its normal state. So I've now told you that HSP47 is really important. There's 2 black boxes. What turns it on and how the devil does it do that? Next slide unveils years of work and tells you the story. Here are the bronchial epithelial cells with the stromal ECM and cells. Chronic inflammation, you induce squamous metaplasia and all the changes. HSP47 is upregulated by TGF-beta. TGF-beta is induced by chemotherapy, carcinogens, any kind of injury, radiation, ROS, anything induces, even think of acute injury, the macrophages that come into that area spew TGF-beta all over the place. What the TGF-beta does is it causes the upregulation of the HSP47 in the fibroblasts, turning them, by the way, into a stiff state, allowing them to enter a stiff state, and that causes an increased deposition of collagen. It causes many things because there's other clients of HSP47, but it causes a stiff microenvironment that can actually work through a variety of mechanisms. I only have limited time, so I'm cutting right to the chase and telling you that HSP47 allows increased collagen deposition, increased tissue stiffness. That stiffness acts through biomechanical signaling to turn on metaplasia and dysplasia. The following experiment just nails that to the wall, because what Dang did is he took the bronchial epithelial cells by themselves and simply put them on gels of varying stiffnesses. So he had a stiffness that was equivalent to normal lung tissue, as well as all the way up to a stiffness that recapitulated tumor, what you see in a tumor microenvironment. And what he found was quite dramatic. As you increased in stiffness, when you went up to the tumor stiffness, you induced the squamous markers and you reduced the bronchial differentiation markers, telling you that simply by putting these bronchial epithelial cells in modulating their stiffness could tell you whether they were going to maintain a bronchial differentiation or transition to a squamous differentiation. Mechanotransduction causes the acquisition of these markers. This works through mechanotransduction, the ability to convert mechanical stimuli into biochemical signals. And this isn't a surprise to people that are familiar with Dennis Dreischer's work from over 15 years ago, where he took embryonic stem cells and he put them on gels of varying stiffnesses. If the gel was stiff, those embryonic stem cells differentiated into bone and cartilage. If the gel was soft, they differentiated into breast, fat, or brain, things like that. At each stiffness, it's an address that codes for a cell identity of an organ. So this is one of the languages that the body uses to dictate cell identity. It works through mechanotransduction. Mechanotransduction often works through YAP-TAD signaling. And so here is a cell at homeostasis. YAP, which is a transcription factor, is located in the nucleus, so it's inactive, and the cell is happy. In a stiff microenvironment, the cell senses that through integrin signaling, which tells the YAP to translocate to the nucleus, where it uses its transcriptional cofactor abilities to turn on programs that are important for regeneration and embryonic proteins programs as well. So this means that if you interrupt this signaling, you should be able to interrupt the induction of mechanotransduction and the subsequent metaplasia. And this is what I'm showing you. Here are the bronchial epithelial cells co-cultured with CAFs. You see the squamous metaplasia. You see the loss of the cilia and the basal cell hyperplasia. So you've induced squamous metaplasia. If you inactivate the signals coming from the integrin, what you find is there is no induction of squamous metaplasia. And if you inactivate YAP function, there is no squamous metaplasia. The cells remain properly differentiated. That means that mechanotransduction working through the integrin signaling to YAP-TAZ and then subsequent transcriptional control is inducing this change in cell identity and the other things that are happening. Ignore the left. It's what YAP-TAZ does. I summarized it for you on the right. It turns on cell signaling. You can see the genes involved. It turns on cell plasticity, so they can change identity. It turns on hippo signaling, which tells the cells whether they're crowded or isolated. It turns on EMT, epithelial mesenchymal transition, and cell migration. It also turns on a variety of immunosuppressive functions. Here I'm just mentioning the PD-L1 induction. So YAP is really important for turning on these regenerative programs that are important during tissue injury. And what I want you to note is that YAP-TAZ turns on all of these things that are needed by cancer stem cells to make a cancer, and actually it turns on all of the hallmarks of cancer. So this regenerative wound healing program turns on everything that a cell needs for getting to metaplasia. And I'm showing you this quick picture. Green is the YAP, and you can see in this area of squamous metaplasia, it's moved into the nuclei of the epithelial cells. Here, right here, I need to get a better picture. Here are the ciliated squamous cells over to the edge. This is just the junction where it starts to turn into squamous metaplasia, and you can see the translocation of YAP into the nucleus. Everything that I've shown you in vitro, you can actually see in vivo. Here is a lung from a person with idiopathic pulmonary fibrosis. We've also done it with COPD. These people are increased risk for lung cancer, and you can see that they have squamous metaplasia. There's the squamous marker. Underneath that is the HSP47 and the associated collagen. Here's a person that smoked but did not yet have lung cancer. All they have is squamous metaplasia, which is quite easy to see in the H&E stain. You can see this robust expression of the squamous marker. There's the surrounding HSP47 and the collagen. None of this is seen in healthy bronchi. And this is an example of a healthy human bronchus. So what I've shown you is that we can generate a functional differentiated bronchial airway in vitro and in vivo. We can cause this to transition to metaplasia and cause it to transition to dysplasia. We can inhibit this transition, and clinically important, we can also inhibit the transition from metaplasia to dysplasia. We've been able to do this not only with these, but with these other model systems. And very interestingly, I've shown you the data from using CAFs from squamous lung cancer, but I can also use CAFs from these other cancers or CAFs from breast, pancreatic, bowel cancers. They all cause the same induction of metaplasia and progression. So the malignant switch is induced by CAFs from multiple cancers. I also have shown you that it's activated early. You can actually injure the fibroblasts and turn on this program. And it's a shared mechanism that's activated early during cancer progression. Finally, how does it provide insight into the immune tumor interactions? What I focused on is epithelial cell consequences of the stiff microenvironment, but the change in force factor affects every cell that's within the tissue. The fibroblasts, the endothelial cells, and least of all, the immune cells. And so very quickly, many people thought that the collagen was creating a barrier for the immune cells to get in, but it's actually much more than that. Here's an effector T cell killing a tumor cell, but if there's a lot of collagen around, the effector T cells are actually suppressed and they cannot kill the cells. Here is a summary of a lot of collagen. See what it does? It suppresses the attraction of T cells, the activity of the T cells. It polarizes the macrophages from an anti-tumorigenic to a pro-tumorigenic state. It does the same with dendritic cells and neutrophils. Here's just a few titles. Collagen density modulates the immunosuppressive functions of macrophages, T cells, etc. And so the CAFs working through mechanotransduction, as well as some secretory pathways, are important in controlling the immune system. And in the very earliest stages, what it sets up is immunosuppression. This is probably biologically relevant because it's important in wound healing. You don't want the immune system going out and clearing out everything. So I want to point you to this last point, that killing the drug-sensitive epithelial cells, whether it's with standard therapy or precision therapy, does not remove the stromal signals that still exist, and they cause immunosuppression in this area, angiogenesis, epithelial proliferation, migration, genomic instability. All of those are still there. You can remove the epithelial cells, but the signals from the stroma are still there, giving it a pro-tumorigenic signal. So mechanotransduction and the stromal signals are involved in the modulation of immunotherapy, and one has to take that into account if you're going to successfully address chemotherapy. The people that were involved in a lot of this work are shown on this slide, and what I wanted to do is leave you with these key challenges for cancer immunotherapy, but I would maintain that understanding tissue biology is first and foremost in being able to manipulate this. Thank you very much.
Thank you for that excellent talk, Thea. Please add your questions to the Miro board. Question. Thea spoke about how the microenvironment influences disease initiation and progression. Where do you think the field currently stands in terms of using those features to kind of predict tumor progression? Specifically, how might systems and synthetic biology be used together to kind of move us close, for using those kind of microenvironmental features in therapeutic design?
So the whole story that I've told you about is how cells deal with stress and either decide to create a protective patch or go forward towards malignancy. And we have markers that indicate that. And when you introduced me, you mentioned that we had developed a clinical test for DCIS, and that looks at stress signals. And what it says is, what we did is we said, look, all the cells of the body are stressed. How do they deal with it? If you look at a biopsy, a DCIS biopsy, and it still is maintaining those barriers to injury, then that cell is not going to progress to cancer. But if it's corrupted some of those responses to stress, that has a high probability of going on to a subsequent invasive cancer. That turned out to be a home run. The epithelial cells that don't properly deal with stress go on with very high probability to form invasive cancers within the next 10 years. We found the corresponding signals in the stroma. So now we have a clinical test. It's being used worldwide. It's reimbursable by Medicare and Medicaid, and it allows one to distinguish at the pre-cancer stage which women are going to get cancer and which are not 10 years before it forms. So I would maintain that we can do this. And what I would like to do is extend the DCIS test to the esophagus, the bowel, the lung, etc. I think it can. I know it can be done because we already have the markers.
Amazing. I guess there's a lot of this has some temporal component to it. Given that how one of the questions here is given how common and rapid processes like inflammation are, what kind of accounts for that long latency in cancer initiation beyond mutation accumulation? Are there rare non-genetic events that constrain progression, and what might these events be?
Well, so acute inflammation does not increase cancer risk, but chronic inflammation does. So the word chronic says that you need time. That's the first part of time. Then once you turn on chronic inflammation, what you need to do is to have the increased stiffness in the matrix. And that takes a little bit of time in and of itself. And so that adds to it. And now what happens is you have a little tiny bubbling point. It's protected, it's immunosuppressed, right? Because it's an area of chronic injury. And now what you have are the makings for both the stroma and the epithelium to allow the outgrowth of the incipient cancer and progress from the precancer to something that you really have to worry about. I hope that answered the question.
Right, and you also talked about a lot about fibroblasts. So there are so many different flavors of fibroblasts. You talked about, stress-related fibroblasts. Do you have any insights on which calf phenotypes might be most important in kind of this driving early disease?
Actually, the marker for these calves is the upregulation of HSP47. If you have any kind of a fibroblast, you can even have a pericyte, etc. If it has upregulated HSP47, it's going to allow the increased deposition of collagen, and you're going to get the acquisition of a stiffer matrix that's going to grow stiffer and stiffer and stiffer. Once it crosses a threshold, then you've turned on the mechanosensory signaling in that area. Any immune cell that crawls into that area, it immediately enters into a stiff microenvironment, turning on mechanotransduction, turning on these pathways.
All right, thank you so much. We have a lot more questions in the Miro board if you could take a look. Unfortunately, we are right on time, so I just want to thank you once again for this excellent talk and move on to our very first session of conversations.
Thank you.
Thank you. I am delighted to introduce Dr. Karin Pelka. She's an assistant professor at UCSF and investigator at the Gladstone Institute. Dr. Pelka's research focuses on uncovering how immune cells coordinate with human tumors to shape immune responses, leveraging log-scale single cell and spatial proteomic approaches. Welcome, Dr. Pelka. We are really excited to have you moderate the first conversation.
Thank you so much. And wow, what a beautiful keynote presentation. That was really exciting. Well, we have two also really exciting talks in front of us, and it's my pleasure to serve as the moderator for the upcoming session. We have Dr. Aventia Rosas-Torres and Dr. Rogelio Hernandez-Lopez. We will kick it off with Dr. Aventia Rosas-Torres. She is an assistant professor of medicine in the Division of Medical Oncology at Keck School of Medicine at the University of Southern California. She's a physician scientist with a research focus on breast cancer and specifically tumor immunology and immunotherapy. She's co-leading the Tumor-Immune microenvironment program at the Norris Comprehensive Cancer Center and runs her own NCI-funded translational research program that is aimed at understanding the role of innate immunity to find ways to reverse immune escape and improve responses to checkpoint inhibitors. And she will now tell us how spatiotemporal biology of tumor-immune interactions can uncover mechanisms of response to immune checkpoint inhibition in breast cancer. So welcome, Aventia.
Thank you so much for that introduction, Karen, and a really lovely talk, Thea. It's really wonderful to hear you speak again, and this is really a pleasure to be here. So I'm going to just jump in because I want to make sure I get through my slides. So I'm going to be discussing immune checkpoint inhibition in breast cancer. And so I wanted to give everyone just a little bit of a background. Okay, sorry, there we go. So this is like very broad strokes, and I know most people on this call don't need an introduction to immunotherapy, and the three main categories of immunotherapy being adoptive cellular therapies, vaccines, and immune checkpoint inhibition. I'm specifically focused on immune checkpoint inhibition in breast cancer, and what I wanted to point out about this particular type of immunotherapy is that it's not as successful in breast cancer as it is in other types of cancer. And there are a lot of different reasons for that. One of them has to do with the intrinsic mechanisms of resistance. And part of what I work on in my lab is understanding specifically immunosuppression as one of these mechanisms of intrinsic resistance. In addition, there's really not a lot of efficacy across breast cancer subtypes. So primarily we can only use checkpoint inhibitors in triple negative breast cancer. So there's a lot more patients in whom we could reach if we could figure out how to get this working better. And then of course, as many struggle with in terms of, predictive and prognostic biomarkers, breast cancer is not unique in this regard, but we do need better predictive and prognostic biomarkers for use for immune checkpoint inhibitors. So one of the mechanisms of intrinsic resistance that I'm going to focus on primarily, and what most of the work in my lab focuses on, is these high levels of immune suppression that can be found within breast tumors and breast tumor metastases. And the main cell types in which we really focus on include the myeloid-derived suppressor cells, T regulatory cells, and tumor-associated macrophages, of which most are very suppressive within these breast tumors. And so the strategy that we are, adopt to overcome this intrinsic mechanism is to really try and focus on targeting these immune suppressor cells as a way to then allow an anti-tumor response to be more effective and activated by immune checkpoint inhibitors. So if we can find a way to either get rid of these immune suppressor cells or change their function, then perhaps we can get better activation of an anti-tumor T cell response with checkpoint inhibitors, which is one of their main mechanisms of action. So the approach in my lab is from mice to men with some math and back. And this is really why I think I was asked to kind of discuss this in the context of this workshop. Is really, this is a systems biology approach, and how are we using systems biology to help us with answering this question. So to start with the mice part of my work, this is just a really brief overview of a lot of the work that was done leading up to one of the projects I'm going to talk about, which is looking at epigenetic modulators as a way to target these myeloid-derived suppressor cells within the breast TME to improve checkpoint response. So the preclinical models showed that when you combine this epigenetic modulator, specifically the HDAC inhibitor, histone deacetylase inhibitor, and tinostat, which is a class 1 HDAC inhibitor, you can really target these myeloid-derived suppressor cells. And when you give that particular drug with dual checkpoint inhibition, anti-CTLA4 and anti-PD-1, you can really see a much more robust response in these preclinical models of breast cancer. The first study really focused on triple negative mouse models and showed that you were decreasing infiltration of these myeloid-derived suppressor cells with entinostat. And the second set of studies showed in a HER2 positive model that you're actually decreasing immunosuppressive function of these MDSCs to allow an improved response to checkpoint inhibitors. That was enough to really lead the way to a phase 1b trial of this combination. And what we did was the first trial was in 35 different tumor types and in the metastatic setting, which showed when you combined entinostat with dual checkpoint inhibition, you got it was safe, and there was a 16% response rate. However, when we went into a dose expansion, specifically in advanced breast cancer, we found a much better response. We had an objective response rate of 40% in triple negative and 10% in hormone receptor positive. Albeit this is a very small trial. This is a phase 1b, not really powered to measure efficacy, but this signal was very promising in a heavily pre-treated cohort of patients, really leading us to try and understand more about what are we doing with this combination. If we understand more, perhaps we can do better, or actually know which patients we could focus on to try and do a larger study and really improve response rates. So the question we asked next was, is modulation of suppression enough to affect survival? Is it really the fact that this epigenetic modulator is doing something to the immune suppression to allow a better response? So this is when we turn to math, and this is the systems biology approach that I kind of undertook with my collaborator, Adam McLean, who's also at USC. And we used a math model and developed this together to identify changes in MDSC suppressive function to see if we could, mimic the effects of MDSCs, the effect of MDSCs on cytotoxic T lymphocytes, CD8 cells specifically, and NK cells, which are known to be affected by MDSCs as well. Can we actually do this enough to result in tumor shrinkage? So this math model was derived to ask this question, and we did this in 2023 and tested it on published patient cohorts for clinical trials, and indeed found that when you specifically modulate MDSC suppressive function on the CD8 T cells and on the NK cells, you do see a significant shrinkage in tumor. So we wanted to know what other cells contribute to this response. So we have this model. If we can maybe understand what other cell types we might need to account for, or perhaps modulate some of the parameters in that model, maybe we can get we can try this out in a larger cohort, and even study this prospectively in our planned phase 2 trial. So what we did was we went back to our mouse models and asked, we use single cell RNA-seq to look at other cell types that are contributing to response. So this is single cell RNA-seq of mice treated with the entinostat nivo-ipi, which is the dual checkpoint with the HDAC inhibitor. We isolated all these different cell types using the single cell RNA-seq and asked about what are the different chats that are affected by this treatment, and could that perhaps help us understand what else is driving response? So which cells are the chattiest, and who's running the show to really lead to a response? So what this analysis showed us is that it's the macrophages, in addition to the MDSCs and the T cells that are chatting, that are the most significantly affected by treatment, in addition to NK cell, MDSC, CD8 T cell chats. So these circuits of chats seem to be the most significantly affected by treatment and perhaps are the driving force in the response that we see. So then we asked which ligand receptor pairs and which pathways might be driving these significant changes. And what came about was that this chemokine, ICAM1, and galectin pathways are driving these myeloid chats. And when you actually go in and look at the LR ligand receptor pairs that might be most responsible for these changes, you can actually isolate specific ligand receptor pairs and see if they're driven by checkpoint or driven by the HDAC inhibitor. That led us to then ask about functional changes. So once we identified these ligand receptor pairs, we went to our immunologic functional studies and looked at changes in suppression of T cells by macrophages and MDSCs by blocking these specific ligand receptor pairs. And what we found was that macrophage inhibition of ICAM1 actually does promote T cell proliferation. And so when we went back to our mouse models and asked, okay, well, is that enough? Can you replace entinostat with perhaps blockade of ICAM1? And what we saw was that, in fact, that's not enough. It's not sufficient, really. You really need whatever else entinostat is doing. So this might be one contributing factor, but it's not the whole ballgame. So at least this did help us to ask, okay, what else can we understand about what's going on in this very complex system? So after we found this effect on ICAM1, we asked, could we go back to our patient samples and use imaging mass cytometry to look at investigation of spatiotemporal interactions to validate some of these findings as a contributing mechanism to response? And what we actually found is in one of the patients who responded, when we looked at where in space in these particular patient responders can we find macrophages and T cells? And does it change over time? Does that maybe give us a hint and kind of corroborate what we're seeing in our mouse models? And in fact, what we saw was that when we did a nearest neighbor analysis, we saw that the CD8 T cell macrophage distance does actually lead to a decreased the distance changes in responders over time with treatment. And because of that change in distance, that actually is very similar to what you would expect to happen when you have decreased ICAM1, which is what was predicted by our preclinical models. So the preclinical model and the mathematical modeling as well really helped us identify that at least this change in ICAM1 pathway signaling between these specific cell types in the TME are likely to contribute. And when we looked in our patient samples, in fact, we did see this. We never would have known to look specifically for this interaction or would have been able to explain how this could contribute to response without incorporating that preclinical modeling as well as the math modeling. And so we then used the parameters that we learned from these experiments and we informed the model, and this validated that the model actually does similarly predict in our particular clinical trial that these changes are necessary to these changes to that pathway are in fact necessary to achieve CD8 inhibition by MDSCs, as well as the need for CD8 activation to cause tumor cell elimination. The model actually really was able to capture this when we ran it, incorporating these parameters that we had learned about. So the take home here is that the use of traditional preclinical modeling and immune assays can be integrated across mathematical modeling and synthetic biologic approaches to accelerate discovery. And I didn't really touch on the synthetic biology that we're actually utilizing, but what we're doing is using patient-derived organoids as a synthetic biologic approach to try and test some of these mechanisms that we are learning about through the data I just showed you. So these are the acknowledgements for the huge team that is necessary to do this work, and I'm very happy to take questions.
Thank you so much, Evanthea. First of all, thank you so much for doing this incredibly important translational work. I'm sure there are lots of questions, but actually the questions will be routed through this collaborative platform. So for now, we're going to move on to the next talk, but please check out this collaborative network to answer all the questions from our listeners. So we're going to move on to the next talk first, which is from Dr. Rogelio Hernandez-Lopez, who is an assistant professor in the departments of bioengineering and of genetics at Stanford University. He's also a Chan Zuckerberg BioHub investigator and a member researcher of the Park Institute for Cancer Immunotherapy. Rogelio received his PhD in chemical physics at Harvard University and performed his postdoctoral training at UCSF, where he was a fellow of the Cancer Research Institute and UC Mexus, which is a UC systems-wide cancer collaboration with Mexico. Rogelio's work focuses on engineering novel T-cell therapies for cancer treatment, and this is also what he's going to talk about now. So welcome, Rogelio, and we're really looking forward to your talk.
Yeah, thank you, Karen, and thank you so much for the organizers for inviting me and the opportunity to present some of my work. So just to put things into context, my lab is a synthetic biology cell engineering lab, and we are interested in using T cells as cancer immunotherapies. But I'd like to mention briefly that we are taking inspiration of how the natural systems work. So a lot of the approaches in synthetic biology try to draw or use principles and from how the endogenous systems work, and then try to mimic or recapitulate some important aspects of it. So in the context of effective anti-cancer immune responses, the initiation of it relies on antigen-presenting cells and the capture of a specific cancer-derived signals, antigens, that are presented later to T cells to induce an effective immune response that relies on the specific recognitions of these T cells to particular antigens. And today I'm not going to talk about other very important aspects, which is the communication between different immune cell types and the establishment of tertiary lymphoid structures to maintain this effective immune response. But what I like to do is to tell you more about how, using synthetic biology approaches, we can mimic this tumor-specific recognition, which is very important for developing effective immunotherapies. So we use synthetic receptors, and I'm sure that you have heard about chimeric antigen receptors. It's a way to bypass the restriction of peptide MHC, but instead using these synthetic receptors to recognize particular proteins that are expressed on the surface of cancer cells. And in this way, we can redirect the powerful killing activity of cytotoxic T cells against cancer. This approach has worked really well in the context of blood cancers, lymphoma, leukemia, and there is a lot of excitement of trying to use similar approaches in the context of solid tumors. However, in the context of solid tumors, there is very few proteins or markers that are tumor-specific. A more common scenario is one where cancer cells express a high abundance of the marker, but those markers are also shared with normal tissues. For example, HER2 that we just heard about in the context is highly abundant in breast, ovarian, colon cancer, but it's also present in a variety of normal tissues at lower levels, for example, the liver, normal breast cells, or the lungs. And that's the case as well of other tumor-associated antigens like EGFR, GPC1, GD2, and others that are currently explored in clinical trials. The problem with the normal or traditional engineered CAR T cells is that although they may have some antigen density sensitivity, it's not enough to provide a safe profile for these approaches. For example, if you try to engineer a CAR T cell against HER2, regardless of the amount of antigen expression, these CAR T cells will kill both cells that are expressing either low levels or high levels of HER2. And as you can imagine, this could be problematic and has resulted in actually reports of what is called on-target but off-tumor toxicity. So some of the work that I have done over the past few years and that we continue working in my lab is using synthetic biology principles to reshape the function and the activity of these T cells so that they have a sigmoidal response, or what is also called an ultrasensitive response, where the T cells only kill cells that express high abundance of these markers, but remain inactive if they find cells that express the same protein, same antigen at lower levels. So today I'm going to tell you very quickly about how you can do that using synthetic biology approaches. But for that, I will have to introduce this new class of synthetic receptors, the synthetic notch receptor, was developed a few years ago by Leo Morsut and Cole Roybal in the lab of Wendell Lin, where I also did my postdoc. And I used the synthetic notch to build what we call an antigen density sensor. So here in what I'm showing you is the synthetic notch controlling the expression of a chimeric antigen receptor. The way synthetic notch works is that upon binding of a ligand to the binding recognition domain, there is a release of a transcription factor, and that transcription factor can be orthogonal and synthetic from transcription factors from the cell, and can be used and leveraged to then control the expression of any payload of your choice. So in these antigen density sensors, what we did was to put the chimeric antigen receptor downstream of synthetic notch. And by playing with the affinities of the recognition domains of these receptors, you can control then the behavior of these engineered T cells. So for example, if we use a synthetic notch receptor of a very low affinity for the antigen, and these cells encounter other cells that express antigens at low levels, because the frequency of interaction between the antigen and the receptor is low, that results in the control of or no CAR expression. However, in the opposite scenario, if these same cells encounter now target cells that express high abundance of the antigen, the frequency of interaction increases, and that leads to the second step and the activation of CAR expression. Because the CAR is a high affinity for the antigen, that promotes what we call a positive feedback. And not only the activation of killing, but also the activation of proliferation and a downstream signaling of the T cells. So to have this overall S shape or the sigmoidal response in the T cell activation. So we have tested this in now a variety of tumor models, but I'm showing you now just a few experiments. For example, if we created a set of cancer lines that have different levels of antigens, and some of them mimic what you will find in ovarian, breast cancer, and also we have some lines that express very low levels, such as those that you will find in normal tissues. And hopefully you can see how these SIG-Notch to CAR T cells have the sigmoidal response that we were looking for. And this is a very different response that what you will find if you only try to modulate the affinity of the receptor, the CAR, if it's all the time expressed on the surface of these T cells. The experiments I showed you were done by flow cytometry, but you could also do microscopy to track the killing activity of these T cells. So before I show you that the traditional CAR T cells do not have antigen density sensing, but these SIG-Notch CAR T cell circuits are very specific to the high levels of the antigen. And here I'm showing you some microscopic experiments to recapitulate that. So now in my lab, we are building the complexity of these circuits and exploring the effect of other parts of these synthetic circuits. For example, the affinity of the transcription factor to the response elements, the costimulatory and signaling domains of the CAR. So there is different elements that can be used to tune the response of these synthetic circuits. And we're also combining this with 3D sphere models and organoid models to both test the efficacy and the toxicity of these circuits, ultimately with the goal of tuning the response and finding personalized T cells with specific behaviors. So but let me move on into second part, just very quickly, just to mention that in addition to this idea of using a single marker to augment the precision of the therapeutic response, you could use antigen combinations, and through computational analysis and other approaches. Now it's clear that we can find sets of antigen combinations that could allow us to increase the precision of the tumor targeting. So for example, perhaps using two different signals, you can design the so-called AND gates or any Boolean gates that will spare the normal tissue. And in work that has been done by Greg Allen and collaborators before, has been shown that using 2 or 3 antigen combinations, for example, you could increase the discrimination between tumors and normal tissue. So in some other recent work that was published just this year, but that I started when I was a postdoc, we put forward this new platform to develop binders. So the one important aspect to build these logic gates is having the ability to have these binders and that are very specific and that can be encoded into these synthetic circuits. So we are using these SH3-derived binding domains that can be engineered to recognize antigens of your choice, and we are calling them Sherpa bodies. And here what I'm showing you is a specificity test where we built a series of URCAT M5 reporters that have chimeric antigen receptors using these SH3 domains against a variety of tumor-associated antigens, for example, HER2 or EpCAM, TROP2, and so forth, and show that you can find some clones that are highly specific for these antigens. And then if you put them together, either as a bispecific chimeric antigen receptor and also controlled by a synthetic notch, you can start building more complex logic gates. For example, you could think about building a tri-specific CAR or a C-notch to CAR where the C-notch controls expression of a bispecific CAR. And here's just some flow cytometric plots to show you that depending on the antigen combinations, you can get the appropriate response for the increasing the precision of the tumor targeting. Work by Greg Allen and others, like Joe Cho and CIMIC and others now using similar strategies. They have shown that they can build, for example, a synthetic notch against EGFR, controlling the expression of bispecific CAR to treat glioblastoma. And this is work that now is, I think, in a clinical trial. And we're really excited about using these synthetic biology approaches to increase the precision of the tumor targeting cancer therapeutics. So with that, I just like to thank my current lab and previous collaborators from University of Helsinki and the funding. Thank you.
Thank you so much, Rogelio. I just checked this collaborative platform and it's very busy. So Evanthia is already answering a lot of questions. You already have a lot of questions there as well. So I hope you have time to interact there for all the specific questions to your talk. We were tasked for the last about 15 minutes now to have a more general discussion of how systems immunology and synthetic biology, how these two fields can really enrich each other. And so I want to start with you, Evanthia, because you already gave me kind of a lead in, and want to ask you how you're incorporating synthetic biology tools. You really mentioned the organoids in your research, and yeah, how you imagine doing that moving forward.
Yeah, so it's a great question. And at this point, so I mean, organoid biology is, still a developing field. And so what I've been doing is I've been working with a collaborator here at USC for the past 3 years to develop a bank of breast cancer organoids, not only early stage breast cancers, but also metastatic from different subtypes of patients. Or different subtypes of breast cancer from different patients. And what we hope to do is to be able to build a platform that we can isolate immune cells of interest from our studies from the patient's PBMCs and basically do 3D type of immune assays in those organoids. So my lab specializes in doing the 2D versions of these. So like immune suppression assays, where you could take macrophages, isolate them from tumors or metastatic tumors, and put them with T cells and tumor cells and ask how this 3 different cell types affect each other. So we basically like to recapitulate that within an organoid, which I only now realize is a huge ask now that I know more about organoid biology and that type of synthetic work. But that is the goal we are working towards. So we are getting closer. And I think what would be really beautiful about that system is that it really does, it can account for some of the heterogeneity that you experience when you look in patient tumors, but would still be controlled enough to at least understand some of the immune mechanisms at play when we're looking at specific drugs and how they affect those immune cell types. So that's kind of our plan. It's a work in progress, but I think we will get there.
Great. So yeah, really using new synthetic biology tools to study the hypotheses that you generate doing your systems immunology approaches. I think that's a really powerful way to do it. I also want to ask you, Rogelio, are you using systems immunology approaches in your research? And if yes, how does that inform your work?
Yeah, no, I think definitely now both systems immunology, synthetic biology go hand in hand. And one way, for example, in which we are exploring similarly to what Vancha mentioned is what are the key elements of the tumor microenvironment that we need to model, no? Or that it will be important to know whether those are sufficient, necessary, for specific anti-tumor or suppression functions. So the idea of incorporating, for example, fibroblasts into these models, or how do you incorporate key aspects of stroma, and not only a lot of the initial assays are only 2D and only cancer cells and T cells, but it is now clear, through several studies, that it's much more complex than that. So we are trying to use this bottom-up approach on reconstituting these microenvironments, either through engineering, for example, cancer cells that also secrete other factors that we know that are important in the tumor microenvironment, and see whether those factors are sufficient to recapitulate what you will find in these patient-derived samples, these more complex in vitro culture models, either organoid derived or synthetic, as I said. I think, for example, the idea of TGF-beta or some of these immunosuppressive signals, you could try to find a cell that secretes those factors and put them into your culture, or you can maybe engineer the cancer cells to secrete those factors, and see if that is enough. So that's the way in which we are integrating some of the systems immunology results and data. And the other, as I mentioned, maybe the computational approaches to know what factors are important, like what antigen combinations will be the ones that will give you higher precision in tumor discrimination, or which signals will recapitulate the specific environments.
Great. So I hear kind of like a guide. What are the problems that I need to solve using my synthetic biology tools? And then also maybe some ideas of how you can build your circuits to solve these problems, like a kind of creative inspiration of maybe what you can engineer in the cells. That's great. Are there any other things just now really open to both of you, and also just like have an open discussions where you see other like the most exciting intersections between these two fields, and maybe connected to that also, like what are barriers that are challenging? We already heard, working with organoids is really tricky. Can you comment more? What are you excited about? What are barriers? How can we maybe solve them?
So I can start. What's really exciting to me as someone who's, really, my background is in cancer immunology and metastasis. And so I consider myself new to sort of systems biology, and certainly, into many of these other platforms. And so I think that what's exciting to me is how powerful they are to be able to really accelerate discovery. So when we come across, especially with a lot of the new technology that allows us to do spatial transcriptomics and, you know, even single cell, which is not really new anymore. As someone who does tumor immunology, it's very overwhelming. We find so many really significant things that are so hard to test in the lab that I almost feel like at this point, it's impossible to actually do this without the help of, you know, synthetic and systems biology to really help us narrow and test hypotheses in a much more efficient way. And so I think that's what is exciting to me, is like learning how to work together across fields to be able to accelerate discovery in this manner. So we can run, you know, my collaborators always say math is quick, or math is cheap. It's not quick necessarily because it's, you know, it's hard to come up with it. But if you want to run 1,000 simulations by just changing a couple parameters that are informed by some of the immunology, you can do that. It's not going to take you, you know, you don't have to, you know, use 150 mice to do that experiment. So I think what's great about that is that we really can narrow in and hone in on what is likely to be really important in the systems that we're studying. And it can really drive us to the answers much faster. And sometimes the answers are, you know, you're not going to find that one target, but these approaches allow you to identify 2 or 3, and perhaps, you know, the HDAC inhibitors, which hit so many of these different cell types, is necessary, and now we can explain why.
Yeah, great. Yeah, so kind of moving from using systems approaches to observe to using systems approaches to really do systematic perturbations in like a high throughput manner, but really with this like mechanistic idea in mind that, yeah, eventually we want to know how all these networks are working together. Yeah.
Yeah, something maybe if I may add that I'm really excited about, and I think it's still a limitation, but it will be very cool, very powerful if we were able to like model from the experimental point of view, like different aspects, that on the tumor microenvironment, having the ability to bioprint, like these hubs of cells, if we are able to reconstitute specific collections of cells with this spatial distribution, also integrating all the information that is coming from systems immunology, but now in these reconstituted systems at a scale, I think that will also accelerate a lot the testing of some of these hypotheses. And I know that the 3D printing, bioprinting, that field is also advancing really fast, and I think that could be also a very powerful intersection with the synthetic systems biology, being able to have these reconstituted systems with very like micron precision or even sub-micron precision, to ask or test some of the hypotheses that come from the analysis. And so that will be kind of the synthetic part. And as you mentioned, the perturbing different cell types at specific time points, I think that also will be very powerful.
Yeah. It's interesting, right? I feel like maybe for a longer time already we had a better handle on cell intrinsic interactions, and I think all these synthetic cell circuits, right, like how they're integrated within a cell, maybe we're pretty good already, and so you can create these new properties of cells. But now having these systems immunology approaches, also understanding how multicellular systems work together, that's now maybe indeed the new frontier where now we again have to come with synthetic biology approaches to be able to model that. And I mean, obviously that's much more complicated, and we heard about the mechanical forces and how that perturbs the system. So yeah, it's going to be really interesting to be able to set up these very controlled bottom-up systems to really understand what's going on. Yeah. Okay. So do you feel there's something that we can do as fields to maybe improve how we're communicating with each other and how, to move that forward more quickly? I think we already touched on it. It's getting so complex. We need to integrate deep mechanistic understanding with like maybe computational approaches to really be able to span this like huge combinatorial space. And then the synthetic biology angle really is yet a completely different set of skills that we need to have. Any thoughts on that? What can we do as a field to move that forward more quickly?
I would say, what I have found to be incredibly rewarding and successful thus far are these really, they're not intricate or complex, but just strong collaborations with folks within these different fields. So, for example, the collaboration that I have with Dr. Adam McLean that I talked about today, and one that I didn't mention today in the slides, but is very robust, is with Dr. Stacy Finley, who's also in attendance today. And, these are PIs who have these incredibly strong, mathematical backgrounds that I know nothing about. And we've been working together as we go. And I think that's kind of the key. Rather than trying to fit, math modeling or agent-based modeling on, onto something that we've already sort of developed in the lab for the way that immunology has played out, we are doing it as we go. So as we discover things, or as we kind of find out, oh, yeah, actually there's 3 different types of suppressive macrophages in the breast tumors, and we find them in multiple models. So instead of having 2 types of macrophages, we're going to have 3, or instead of having 1, we're going to have 2. And instead of calling them M1 and M2, we're going to call them, we're going to give them the different features that we've identified within the breast tumor microenvironment that are super important. And so like working together, over the course of time, I think is going to be the best way to do this. And really, we can then come up with new ways in which we can use them. Oh, maybe we can study this prospectively as part of a correlative in our clinical trial, these kinds of things where we don't wait to kind of retrofit things. We do it as we move forward in our own studies.
Yeah, I think it just continue promoting spaces like this, and through funding mechanism, just meetings and ways in which we can interact more and learn from each other. I mean, this space and fields have been growing so much and are attracting more and more people from different angles. So continue building these bridges and establishing the common languages, and simplifying things so that like the different backgrounds kind of meet at the same level. I think that's very important. But yeah, it's exciting right now to be in this space.
Yeah, I agree. It's a really exciting time. I think we hit the time. There's gonna be a 15-minute break, I think. So there's gonna be more time to look through these like specific questions to your talks and continue the more general discussion. I'm gonna try to wrap it up with my own words. I think what we heard in your talks and also throughout the discussion is that on the systems immunology side, we're really excited that we start to move from profiling and observing variation to really starting to understand it by incorporating mechanistic studies and systematic ways, and that there we really benefit a lot from the synthetic biology tools that are being developed now. And then especially throughout your talk, Rogelio, I heard that for the synthetic biology side, the systems immunology really informs the hypotheses, but then also it's very powerful to use the synthetic biology to move beyond the natural variation and beyond what we see and response and resistance and engineer the properties that we really need to come in therapeutically. And I think this is a really exciting time to do that. And yeah, so I hope there's going to be a lot more discussions. I actually don't quite know. Do I hand it back to someone? I'm not quite sure what's happening next.
Thank you, Karen.
Enter the break.
Thank you so much, Karen, for moderating such an exciting session and thoughtful discussion. We'll now take, I think, a 13-minute break and reconvene promptly at 2 p.m. EST.
Awesome. Thank you so much again.
Welcome back, everyone. I am excited to start our next conversation. So I will now introduce our moderator for conversation 2, who is Dr. Elizabeth Wayne, an assistant professor in bioengineering from the University of Washington. Just for background, the Wayne Lab harnesses synthetic biology and nanoparticles for immunoengineering to study and address chronic disease. So please take it away, Dr. Wayne.
Hi, everyone. It's such a pleasure to be here. I am, very excited to be here. So I want to introduce our two main speakers for this session, Dr. Jose Reyes and Dr. Xiaojing Gao. Our first speaker, Dr. Reyes, is a postdoctoral fellow at Memorial Sloan Kettering Cancer Center, and he is jointly working between Scott Lowe and Dana Pierre. He works to understand how direct subpopulations of cells and tissue respond to genetic alteration of oncogenes and tumor suppressors. So very excited for his talk. We also have with us Dr. Xiaojing Gao, who's an assistant professor of chemical engineering at Stanford University. His lab tackles fundamental engineering challenges across different levels of complexity. So he looks at protein components and how to minimize their crosstalk in human cells and immunogenicity, biomolecular circuits, and multicellular consortia to communicate across scales, and also therapeutic tools. So I think this is going to be a really interesting talk about synthetic materials, but also cell-cell signaling. So Dr. Reyes is going to speak first, and his talk title is The Making and Breaking of a Pre-Cancer Niche. Welcome, Dr. Reyes.
Thank you so much, Liz. Okay. Thank you so much for the organizers for inviting me to present here today. It's such a treat. I'm Jose. I'm a postdoctoral fellow at Memorial Sloan Kettering in the labs of Scott Lowe and Dana Pierre. And today I want to share with you my work on the making and breaking of a pre-cancer niche during pancreatic cancer initiation. Cancer emerges as a result of a cell state transition in which genetic alterations in otherwise normal cells lead to changes in cell identity and hallmarks of malignancy that ultimately make cancer lethal. However, cancer development is more than this. It's not only about a cell state transition, but a transition in niche state. And pancreatic ductal adenocarcinoma, or PDAC, is a paradigmatic example of a type of cancer, of the type of profound tissue level changes that occur during cancer development. In its clinical presentation, PDAC presents as a stromal desert in which most cells in the tumor mass are actually activated fibroblast or immune suppressive myeloid cells that can both contribute to tumor development and impair therapy responses. How does cancer build its niche, and how can we prevent or revert this niche state transition? I think that in order to ask these questions, we need to go back to the origins of PDAC, and genetically engineered mouse models provide a tractable experimental tool to study this process, because we can engineer them to predispose them with oncogenic mutations that will that will make it really likely that they will develop PDAC later in life, but study them in the very early stages of this transformation process before cancer even emerges, and gain insights into the tissue level, molecular level, and cellular level events underlying PDAC initiation and progression. And a number of years back, members of the Lowe and Pierre labs, as well as other labs, started gaining insights into the spectrum and dynamics of premalignant cells that emerge in these mouse models of PDAC. What I'm showing you here is some example data of single-cell transcriptomes in which each dot is a single cell, and cells are plotted together based on transcriptional similarity. And this data comprises of 1.3 million premalignant cells that have activated the oncogenic KRAS mutant and have initiated neoplastic transformation, but they're not yet cancer. And early work in the lab showed that oncogenic KRAS leads to the diversification of the types of states that premalignant cells can acquire. And in this talk, I will focus on two of these subpopulations that represent two diametrically opposed premalignant phenotypes that epithelial cells can enter. Gastric-like cells express markers of the gastric lineage otherwise absent in the premalignant epithelium. They also resemble benign lesions and retain a certain degree of epithelial identity. On the other hand, progenitor-like cells express nestin, a marker of pancreatic epithelial progenitors. They also engage signatures of the epithelial to mesenchymal transition, and among all premalignant cells are most similar to cancer. These two states have very distinct transcriptional profiles. So we have a set of genes that become upregulated selectively in gastric-like cells and are sort of downregulated or absent in progenitor-like cells. And conversely, there's a set of genes that are upregulated in progenitor-like cells, but absent in gastric-like cells. But I think you can also appreciate that these two are just extremes among an entire spectrum of phenotypes that include a set of cells that have a mixed sort of profile. Using computational tools, we can identify major axes of variation in single-cell data and try to analyze the premalignant spectrum among a continuum between gastric and progenitor-like states. And in doing so, we can reveal that entry into the gastric-like program or progenitor-like program occurs through a gradual induction of a set of genes, suggestive of the type of dynamics that may be occurring in vivo when cells adopt these different fates. What I haven't told you yet is that this data actually doesn't come from dissociated single-cell data, but comes from single-cell transcriptional data that was derived using the 10x spatial transcriptomics Xenium platform. And what this means is that for every one of these 1.3 million cells that we have in this map, we have not only its transcriptional makeup, but also its spatial location in the premalignant tissue. And this seemingly simple new piece of information opens up a world of questions that we can ask about the state of premalignant cells, because we can both characterize them in terms of who these cells are, but also what is the state of all their neighbors in their niche. And here I'm defining a niche as the set of cells that lie within a particular radius, in this case 60 micron, of a point of reference in the tissue. In premalignant tissues, we can identify niches that are rich for gastric-like cells that I'm going to call gastric niche. And conversely, we can identify niches that are rich for progenitor-like cells. I'm going to call this progenitor niche. And also niches that are rich for cells in between sort of these two extreme phenotypes. And because we can order sort of niches in this way, we can now ask, how does the microenvironment around premalignant cells change as we move sort of along the spectrum of the gastric niche to progenitor niche? Playing the same game that we did for analyzing gene expression trends in the sort of epithelial state, we can now ask, how does expression of fibroblasts, for example, change when sort of in the vicinity of cells that are in the most gastric-like state or progenitor-like state? And I think what you can appreciate from this plot is that fibroblasts are very different depending on the type of premalignant cells that they are surrounding. The other thing that you can appreciate is that there seems to be a very sharp transition a sharp transition in the gene expression of these different fibroblast populations, suggesting that the adoption of the gastric-like state or progenitor-like state occurs through a niche state transition. In the case of the progenitor niche, the fibroblasts that inhabit sort of or surround this type of premalignant cells are sort of show signatures of the activated myofibroblast subtype. And this is evidenced by expression of genes such as tennescin C, alpha smooth muscle actin, pyridoxine, or TGF-beta, which have been shown to promote PDAC progression in mouse models of cancer. In the myeloid compartment, myeloid cells have signatures of immune suppression, such as upregulation of PD-L1, and we have also seen other markers that are consistent with an immune suppressive phenotype using reference-dissociated single-cell data. When we look in tissue, these two types of immune and stromal cells localize really sort of tightly with progenitor-like cells shown here in cyan in the premalignant tissue. And strikingly, these type of niches resemble the microenvironments that later go on to develop when mice get PDAC, suggesting that these niches may represent some type of precursor of what's about to come upon malignant transformation. So we naturally ask, can we intercept the progenitor niche before PDAC development? And recent developments in medicinal chemistry have allowed us to do this, because small molecule inhibitors that are specific for mutant forms of oncogenic KRAS, such as MRTX1133, allows us to target specifically the premalignant cells without directly disrupting the microenvironment. I designed an experiment in which I subjected a mouse that has activated oncogenic KRAS in the premalignant in the epithelium, and this is a really young mouse, so this is a premalignant stage of pancreatic cancer development. And I treated these mice with either sort of the oncogenic KRAS inhibitor or vehicle for only 2 days, and harvested tissue at the end of the treatment for histology and spatial analysis. And this treatment had a profound impact in the abundance of progenitor-like cells in the tissue. What you can see here on the left is that progenitor-like cells marked by HMGA2 in yellow are very abundant in the vehicle control tissue, but they almost disappear just after 2 days of acute oncogenic KRAS inhibition. And I want to point out that this this happens without complete ablation of the premalignant epithelium, implying that this progenitor-like population is particularly sensitive to oncogenic KRAS inhibition at these very early stages of pancreatic cancer initiation. And the effects of the oncogenic KRAS inhibitor go beyond just what happens to progenitor-like cells to what happens in their niches. What I'm showing you here is some representative images of tissue that we analyzed using the Xenium platform. And I'm highlighting just the microenvironment cells that are mostly associated with progenitor-like cells. And what you can see is that in the vehicle, these microenvironment cells are pretty abundant, but just after 2 days of oncogenic KRAS inhibition, these microenvironment cells are mostly depleted. The picture that comes up from these studies is that progenitor-like cells emerge as part of a niche state transition during PDAC initiation that starts resembling what a cancer will look like when it eventually develops. And removal of even one of these components is sufficient to dismantle the entire niche, implying that there's a very a tight relationship and interdependency that is built early on between these different cellular communities. One question that I would like to end this talk with that comes from these studies is what happens when we think about tissue remodeling as a dynamical system, and whether self-reinforcing multicellular circuits may be leading to the formation of these really well-defined premalignant niches, but also niches present, for example, during wound healing or in tissue regeneration. And I think thinking dynamically about tissue remodeling has important implications regarding the questions that we can ask. For example, I presented an experiment that looks like this, in which I drove a system that had really high abundance of progenitor-like cells to a state that had really low abundance by a really high dose of oncogenic KRAS inhibition. And I showed you that this had profound implications in the type of microenvironment cells that surrounded these cells. But within this x-axis, we can imagine an entire continuum of different densities of progenitor-like cells. And I cannot help but to wonder, is there a critical number of cells that we have to get rid of in order to lead to the niche collapse? Is there a tipping point in this system? Similarly, is there a critical number of cells that initially have to accumulate in order to build the niche to begin with, a phenomenon known as hysteresis, where we have two distinct tipping points? And in order to understand these questions, we need new tools to dissect the ecology of premalignant tissues. In particular, we need to visualize and model the spatiotemporal dynamics of cellular communities in vivo, and to conduct quantitative perturbations of cell states in a similar manner to how we conduct quantitative perturbations to molecules. So I think a recurring theme of what I'm thinking right now is that cells in some way in these dynamical systems are becoming the new units of analysis and of perturbation. And I would like to keep discussing this in the rest of the meeting. With this, I would like to close my talk and thank Scott and Dana, my advisors, who gave me the opportunity to be in their labs and learn from a lot of really good friends and colleagues, all sorts of stuff like pancreatic cancer models and also single-cell genomics, as well as collaborators in Sloan Kettering and beyond, and the generous funding sources that make this work possible. Thank you very much.
Thank you for that talk, Jose. That was wonderful. Always a pleasure to see, actually, and great explanation from having read the paper. I'd like to introduce the next speaker in our session, Dr. Xiaojing Gao, Assistant Professor in Chemical Engineering from Stanford University. After our two speakers, then we will have a discussion, and I also welcome you to check out the board if you have any other thoughts already from Dr. Reyes' talk. And now Dr. Gao is going to talk about programmable RNA sensors for internal and external states, or his new title. Thank you.
Yeah, thank you, Liz. Good to see you again. I want to thank Lucas and the organizing committee for this great opportunity. Very exciting roster. So yeah, I try to broaden up a little bit, and today I want to share two tools with you that I think could be useful for this community. But let me step back a little bit, saying we are really at an exciting time where it's been shown that it's possible to deliver genetic payloads into patients using RNA vectors. And compared to more conventional DNA vectors, they are safer for these two reasons, But historically, in synthetic biology, I would claim there are many more tools, although there are always exceptions, but there are many more tools that operate on DNA than after the transcription regulation. So, and you can see these kind of DNA level regulation is not really compatible with RNA delivery. So the mantra of my lab is to map this sense response process that we talk a lot about in synthetic biology onto components that either operate on the RNA level or the protein level so that they can empower smarter RNA vector-based therapy. But I also want to point out for this context, it doesn't have to be RNA. There are other nice features of the tools we develop as well. So just to jump into the two tools, first we want to talk about how do we detect extracellular signals. You just heard Rogelio talking about SynNotch, and Josh is coming up in the next session. His lab has a whole body of work making MESA. So we're trying to take this inspiring history of making synthetic receptors, but take them to an RNA-compatible place. And this work has been led by Xiaowei and Santi, and we just walk you through how this works. So in this synthetic receptor called LIDA, it has three components. The first component is a piece of RNA that has three coding parts. First part is the optional marker. The last part is the payload that will be conditional on the input. And the key in the middle part, that's a stem loop onto itself with an in-frame stop codon. So the default is there is no output. The second part is a truncated version of ADAR that eventually would be able to add the stop codon, but they are truncated and not very efficient doing so at the baseline, fused to one dimerization domain. And the last part is the other dimerization domain that always sits on RNA sensors through a specific protein-RNA interaction. And these dimerization domains were chosen such that in the presence of the right ligand, they'll come together. That will bring the editing enzyme into a spatial proximity of the stop codon. And then what this enzyme does specifically is within a double-strand RNA region, it will rewrite I into A. And for cells, A is treated as G. So now stop codon becomes a tryptophan codon, and the translation can go through and make the output, right? So skipping over a bunch of optimization, we did show that it works with actually all kinds of different dimerizations. And one thing that's nice beyond just RNA deliveries, because all operation at the RNA or protein level, the multiple component receptor can be encoded as a single transgene and then transfected into cells. In this case, as a dimerization domain, we chose GPCR and arrestin because we know when GPCR gets activated, its C-terminus is phosphorylated and recruits arrestin. And then we use that to successfully demonstrate a single transcript delivered three components receptor that can respond to a ligand vasopressin that activates a specific GPCR we chose to engineer. And since we talk about RNA, we did also show that you can deliver this system using in vitro transcribed mRNA as well. In this case, we built a sensor using a different GPCR that responds to a different ligand. In this case, it's a chemokine CCL20 that's actually elevated in some tumor microenvironments. And in a glass half empty, half full situation, we were able to show that it is still compatible when you use these commonly used modified bases that people use to reduce the toxicity and immunogenicity of in vitro transcribed mRNA. It's reduced, it's still working, and we're actively thinking and exploring how to bring these editing efficiency to the same level as unmodified RNA. But suppose one day we could make that, one thing we could do is to make a more specific in vivo CAR T generation using RNA, just the same way SynNotch has revolutionized how people do lentiviral-based ex vivo CAR T engineering. So imagine you can deliver mRNA using lipid nanoparticles in vivo into T cells. That was first make a LiDAR sensor, and the LiDAR is programmed to detect markers that's specific to a tumor microenvironment. And then only when these engineered cells are in the right microenvironment, they will start to make make CAR to target their target cells to ablate, to avoid off-target ablation in other tissues. That's quite common in this kind of therapeutics. And also, we've heard about a lot of inspiring talks about the niche. We are obviously not there yet, but in a synthetic situation, we're asking, can we use LiDAR to detect different kinds of different physical form of ligand? So we play the sender cells, the blue cells in the middle, and we played the receiver cells that makes all the LiDAR components on the outside, that's the red cells. And the sender cell either makes the ligand that diffuse out towards outside, or the ligand is tethered on the surface of the sender cell. And so in these cases, you expect different spatial distribution. This signal will go out, this signal will stay on the border of the sender receivers, as you can see here. And it probably is easy actually to see on the quantification. So this blue line is where the sender cells are, the red line is where the receiver cells are. And in the pterodactyl case, indeed, the green signal is limited to where these two populations touch each other. Whereas when we have a diffusible ligand, the signal clearly goes out beyond where the sender cells stop. And perhaps one day this could be used to visualize all these local communication cues in live animals or in mixed cells models of tumor development. Now, switching gear to the other end of the equation, we also want to control intercellular communication. And skipping over a bit of background, but basically back when I was a postdoc, we figured out that you can take some of these proteases and regulate them to talk to each other and process signal. We figure out between us and the field, there are many ways to convert pathways of activity or pathways of interest into protease activity and feed them into the circuit. But what was missing is back then the work can only use to regulate cytosolic proteins. We didn't have a good way to regulate secreted and cell surface proteins, which are important, again, in this context. So when Alex started this project, we decided to hack endogenous pathway that's responsible for this. So now you're looking at the bilipid layer of ER membrane. This is the luminal side that will eventually face the extracellular side, and this is the cytosolic side. The cargo is fused through a transmembrane domain to a retention signal that tells the cell to keep the whole thing in ER. But now, in response to a protease activity that will be coming out from the circuits I talked about before, you remove the retention signal and the rest is free to transition onto the Golgi and beyond. At this point, though, this cargo is still tethered to the membrane. So to free it from the membrane, what we did is we have a second protease cleavage site for endogenous protease furin. Furin is present only in Golgi, but not in ER. So only after you have the first event, the second event happened, and end result is if you can control the protease activity in the cell, you can control the protein secretion of arbitrary proteins. And Roman Girard's lab, actually our paper came out to back develop a very similar system for this purpose. And let me just give you one example of what we were trying to do. So we are on the notorious oncogene KRAS. We know people try to inject IL-12 locally into tumor preclinical models as a potential therapy. And we asked, can we build a protein circuit that can detect KRAS and then make IL-12? Let me walk you through. So first, the sensing part. In the sensing part, we take a protease, split this into two halves, each fused to a binding domain that binds to active KRAS. So by default, these two halves will float around in the cytosol and nothing happens. But when there's active KRAS sits in the membrane, both will be recruited to the membrane. You transition from 3D diffusion to 2D diffusion and increase their effective local concentration. And because the affinity between the two halves, the protease are non-negligible, that transition seems to be sufficient to reconstitute some protease activity. And the second part is less of a process, more like a relay, because this input protease is reconstituted on cell membrane, but the release mechanism I just told you about happens on the ER membrane. So we have a relay protease, half of which is already sitting on the ER membrane. The other half starts on the cell membrane with spatial sequestration, and also the activity is sequestered by using the dead half of the other protease. But you have these cleavage sites for the input protease. So once this protease is activated, it will cut off here and here. Now this part of the relay protease is free to diffuse away, and some of them will find their way here. And the last step is the mechanism I just told you about to control IL-12 secretion. So this is all toy models, right? Not real cells, HEC293 cells, either baseline or co-transfected with a KRAS mutant. And at least in the artificial context, we were able to increase IL-12 secretion in response to KRAS mutant. And what we have been doing since is to really trying to make our protease toolkit rival the capability of transcriptional toolkits, such as sometimes you want to flip the sign of control, sometimes you want to tune the sensitivity to inputs or the amplitude. And we actually figured out all ways to do that. Just seeing one last data slide, what we realize is this seemingly boring, unremarkable transmembrane domain is just a single pass alpha helix, right? But we tried all the transmembrane domains we can find in the human proteome and realized that they have huge impact on how our system works, right? So y-axis to the right is better, tells you how strong the surface expression is. So that's x-axis, and the y-axis tells you how responsive it is to our ER retention regulation module. It turns out that our favorite transmembrane domain sits somewhere here, and we have much better candidates these days sitting here. And this also gives us more data to mine, because there's all these non-trivial patterns, and we are now trying to write machine learning algorithms to figure out, these similar transmembrane domains, what features are there to determine their responsiveness to ER retention regulation and the expression level on the surface, and how do we use that to actually predict the consequence of some of these clinically observed variants and how they will affect protein secretion. So that's all I have. These two tools I share with you today, some other tools we build in the lab. And most importantly, if any of you think any of these tools are useful, please always try them out. And if anything is not working as intended, please email us. As tool builders, there's nothing more exciting than helping users figure things out. And with that, I just want to thank the lab and the funding sources that made it possible. And I think we will move on to the next panel discussion. Thank you.
This is awesome. Such wonderful talks. Thank you. Always a pleasure to hear you talk, Xiaojing. And good to see you too. So I also saw that our chat board was also very active, so I'm very excited about that. Keep that going, and I'm sure Jose and Xiaojing will be able to check in that later. But for now, I do have some questions that I will kind of alternate between you guys or for both of you to answer. So I'll start with something for you both. And this is also drawing on some of what you talked about in some of the most recent papers you had. But what I thought was interesting is that both of you involve approaches that are post-transcriptional for communication and signaling, whether it's in Dr. Gao's case trying to modulate or actually do therapeutic interventions with cell signaling, or actually epigenetic understanding that these cell signaling modifications are very important. So can you really spell out for me the significance of doing post-translational modifications, or what these relevance are the post-translational side and post-transcriptional side, I would say, apologize, post-transcriptional versus this previous maybe inference on just pre-transcriptional?
Maybe I can comment on this. So I think maybe I can turn the question more into a challenge. So I think the types of cell-cell communication circuits that we're studying sort of involve diffusible ligands, right? That are sort of proteins themselves. So, but I work sort of on a technology that relies on inference of this type of communication from transcripts. So one challenge that we have in the field is how do we understand sort of from the expression level that a cell has, sort of how far away are sort of is a signal inferencing other cells, and how can we get a sense from the transcriptome as to whether sort of there's any functional interaction between cell states? And I think this is a question in the field that is actively being studied, but sort of from a qualitative perspective and a quantitative perspective, and I think is still unresolved. So that's maybe something that I think.
Awesome.
Yeah, and actually a point related to Jose's point, and I may be leaning too much into a therapeutic angle, but the most immediate application of our release platform, for example, would be such, right? Because both in his talk and in the previous session, we saw that there's all these intercellular communication molecules that's probably involved, right? And Jose also made a very nice point. You want to have a quantitative tuning. So imagine how would you quantitatively tune, say, IL-2 without interrupting its endogenous regulation. You can't just go in and put like a doxy-inducible promoter on IL-2 because that will interfere with the other regulation that's also happening there. But what we could do, what we could envision, is you take this little retention tail from release, knock this into the C-terminus tail of your favorite secreted factor, and then also co-produce a small molecule inducible proteins. Right. So what that will allow you to do is preserve as much as a natural regulation of that secreted factor as possible, and then dial in to get different level of that, a different strength of that specific connection. And I suspect in that case, you might get more nuanced answers than just they knock out that or block it with antibody. So that's one thing. I've been trying to pitch that for people who care about these kind of problems. If anyone want to give it a try.
Yeah. And that's probably, do you think that's something that's better regulated at the RNA level? I mean, you might say yes because you've already got technologies for RNA.
So we don't know, right? But in this specific example, if the cell also regulates RNA of these secreted factors, we are not touching that either. So we are preserving that as well. We're just trying to put in a tuner.
Yeah, To go back what you were saying, Jose, about this, the need for spatial resolution and the questions that brings up. When I see spatial data, I'm always fascinated by it because I think there's a difference between what kind of spatial resolution we can achieve, which is in engineering modules, you like to engineer gradients of change, and then what matters in terms of the spatial engineering when you're studying these kinds of cell-cell interactions. So I'm kind of curious, like, while you mentioned it's still challenging to get that kind of understanding, but I'm curious if you have any ideas of what level of spatial resolution you need for these kinds of signaling. That I think would be useful for people who are trying to develop strategies to then achieve that spatial engineering, that spatial resolution.
Yeah, and I think this is a great question. It's something that we're actively thinking about. And maybe, for example, in my talk, I sort of brought about this idea of defining a niche as a set of cells that lie within a particular radius that I chose arbitrarily, right? It's 60 microns. But sort of something that comes up when doing this computational analysis is how do you define sort of that length scale, right? Why not 30 microns? Why not 100 microns? And I think the truth is that sort of different patterns in sort of tissues are operating over very distinct length scales. So I think maybe I advocate for sort of both a holistic sort of analysis of tissue heterogeneity that considers all sorts of different length scales, and also sort of for the role of observational approaches for informing sort of synthetic biology approaches and perturbations that will recapitulate the type of natural length scales that are operating in living systems.
Yeah, no, it's challenging because there's no simple way. You need both. Cells both communicate on their neighbors, and there's also across tissues or across actually even organs.
Yeah.
Something I also thought was interesting. I guess let's say you guys had to write a grant together. Because one thing I thought was interesting is you have someone with all of this data looking at these cell-cell interactions and being able to take a mutation and go, when you do this mutation, here are all of the ligand interactions, the receptor interactions that are changing. And then you've also then just beautifully been arranged to talk with someone who can specifically design synthetic receptors and looking at those very kind of outputs. So is it that easy? What are the kind of is that a connection that makes sense as experts? Or what are hidden challenges for how you merge these both fields of thought and tools? Hopefully that was a good question. And hopefully someone funds you to do this work.
I don't know, maybe I could share the challenge when we try to be the vacuum salesman and pitch our tools to people. It's actually very hard to find a perfect application where people have to watch this in live, right? Because what our tools would allow you to do is you let the animal do its thing, and then you can analyze them later and figure out historically what was the signaling event, or even maybe watch them live, right? But it turns out that with these spatial transcriptomics, you can already learn so much, and you can do like a pseudo time. So in most cases, it's like we could not convince people that actually you need to do all the extra transgenics to get a little bit more information than what one is already getting with all the nice tools that people have on fixed dead tissues, right? So that's a first. And the other thing is related to that is also the technical barrier is still a little too high, because we try to make it a single transcript, right? Easy delivery, but still in most cases, people are like 2 transgenes is too much, even 1 transgene is too much than 0 transgene. So these two things are linked together.
Sorry, I turned the video off, but I can still hear you. Mother got a kidney transplant yesterday, and I'm in the hospital. I know sorry, that's not relevant to here, but it's kind of a big deal waiting over a decade for a kidney, and it finally. So anyway, I quickly changed my plans, but keep talking. The video might come out, but I'm here. And thank you for anyone who does kidney work on this call. Please finish the question. I really want to hear what you have to say, Jose.
Yeah, no, so what I was going to comment is that I think that sort of bridging this gap and sort of having the ability to perturb sort of the systems quantitatively is going to be really important to understanding sort of to really bring sort of home the perspective of tissue biology as a dynamical system. I think that the tools that we currently have are really blunt, and I think that sort of doing these quantitative perturbations is going to be really important to sort of understand these input-output functions sort of in tissues in the same way that sort of the field of systems and synthetic biology have done so for understanding molecular circuits.
Okay. This is interesting. Thank you so much. I'm always thinking about therapeutics. And so my question, hopefully you can still hear me, Jose, a lot of your work talked about initiation of pancreatic cancer. And I'm kind of curious if someone were thinking about how to develop treatments or cancer, like diagnoses, what does it mean for discovering something initially, particularly when maybe someone discovers they get a diagnosis that's far later?
Yes, so I think like maybe we're actively thinking about this, and I think it's a really important consequence of the type of work that me and other people are doing. And I think about this in two arms, right? Like either sort of the diagnostic perspective, which is more sort of on the observational side, and then the intervention perspective on the perturbations. I think this question comes a lot. Like if you think of a pre-cancer, and I think Thea gave a really nice example of how we can use this for diagnosis. But I still think, oftentimes I get the question like, is there any secreted molecule that becomes selectively activated in this pre-cancer state that you could detect, sort of, for example, in a blood test without some invasive procedure? And I think maybe sometimes there's definitely secreted molecules, but I don't know sort of if the really, really early stages will sort of generate sort of critical mass of these molecules that will be sort of reliably detectable, or sort of these will be detected sort of only when progression has already advanced pretty far along. In terms of intervention.
Can I ask you a different question then, or a related
Yes.
question? Because I guess I want to say I believe you, and I think this question is worth answering for answering its sake, right? I think there's value in this, and everything. Not every development has to lead directly to a therapy. But my curiosity here was about whether you thought if you did the same model for something that was late stage, is it that it would be harder? Could you even do this kind of analysis? Because maybe the mutation or the interactions are going to be far more complex and far less easy to define. Like given what we know about early stage and immune escape, right, and that happens, the elimination phase versus the immune escape, is it really harder to do pathways that are late stage versus the initiation? Like they're kind of
Yes.
analysis that you do.
Yeah, so I think in terms in pancreatic cancer, in terms of genetics, so it's something really interesting because people often say in the field like pancreatic cancer has like really simple genetics, at least initially, right? So it's most of the cases start with oncogenic KRAS activation, and then sort of there's three main tumor suppressors that become sort of recurrently mutated in the path for malignancy. Whereas, for example, when a tumor develops in a p53 deficient context, sort of tumors become genetically very complex, and there's also sort of more perhaps opportunities for interconvention between states and heterogeneity. So I think maybe the complexity of the pre-cancer, I think, is probably much sort of smaller than the complexity of the cancer. And I think there's a lot of opportunities for early interception of these targets. And I can also share with you that sort of some experiments that I didn't present because they're very kind of preliminary, but I am sort of doing proof of principle experiments of what would happen, for example, if we were to treat a mouse with just 3 days treatment of oncogenic KRAS inhibitor really early on. Is this sufficient to delay tumor progression or to prevent it, right? And I think maybe in terms of in pancreatic cancer, this has a good rationale just because most of the cases start with sort of predominantly with mutation in these particular oncogene. So I think there's a lot of opportunities for thinking about interception of early disease especially if there's some predictability to the fact that these may be present, these mutant cells may be present there.
Thank you so much. Thank you for tolerating my absence of video, the hospital announcing they had a discount somewhere. And I had one more question for you, Xiaojing, but out of respect for you as speakers, and I think the next panel that's coming on, I'm going to hold my talk because I know I can email you. Thank you all so much for a wonderful presentation, a wonderful discussion.
Yeah, no, thank you so much. And I hope your mom, everything goes well with your mom.
Yes.
Thank you all once again for this, wonderful conversation. Let's move on to the next session. So it's a pleasure to introduce Dr. Tudia Bruno, Assistant Professor of Immunology at the University of Pittsburgh and a faculty member at the UPMC Hillman Cancer Center. Dr. Bruno is recognized for her work on B cells and tertiary lymphoid structures in solid tumors, with a focus on understanding how these kind of structures shape immune responses to cancer. So welcome, Dr. Bruno. We are thrilled to have you moderate this discussion.
Thank you so much for the invitation to be here today. I appreciate it. And all the talks have been super wonderful and the discussion has been super wonderful. I have the pleasure of introducing the first speaker of our session. And so actually, I just realized, is it going to be Lucas or Josh going first? I didn't know if we changed the order. So sorry for that.
I can chime in. I think we're planning to start with Lucas.
Okay, that's totally fine. So, it's my pleasure to introduce Dr. Lucas Pelkmans, and he's here joining us. Hey there, and he's gonna share his screen while I just do a brief introduction of him. So, he is currently assistant professor at the Institute of Molecular Systems Biology in Zurich, and is going to be talking to us more about thinking about multimodal perception of individual cells and linking that to cellular state of drug response. And so, Lucas, I won't take any more of your time. We're really looking forward to your presentation. Thank you.
All right, thank you very much. I hope you can hear me well. I'll share some of our research that really is about looking how individual cells respond to growth factor and how they do this in a contextual manner. And the technology that we use for that is called 4i. It's a technology we developed a couple of years ago in the lab, which allows us to really achieve highly multiplexed staining of cells and tissue samples across multiple spatial scales. So we can get very high spatial resolution imaging of up to 100-plex protein state stainings, all the way up to millimeter-sized samples. So that really allows us to extract a lot of information of individual cells within that context. And the data that we generate with that is really powerful in combination with computer vision and machine learning approaches. And for the sake of this talk, I would like to introduce two concepts that are actually very well studied in cognitive sciences, but not so well studied in our world of cell biology and cancer biology. And the first concept is called contextual information processing, and I would like to illustrate that with this slide. So when you look at this slide, you see two identical objects or symbols. But if I show it to you in this context, you will very likely read one symbol differently as another symbol. So this is, of course, a perception generated by our brain, by a multicellular network, and that allows the same input to give a different response, but also it can sometimes allow different inputs to give the same response. And one of our hypotheses has been that actually individual cells can very much do the same contextual information processing using molecular networks. So even though it's a very different spatial scale, molecular networks may also have the ability to do contextual information processing. So to ask this or to study this, we applied our 4i technology, and we used that to multiplex multiple signaling activities in single cells. So the active states of multiple signaling nodes in a typical growth factor response network, as illustrated here. But then we also multiplexed in the same cells multiple markers of the cellular state. And so these are markers related to the cytoskeleton and the mechanical states, also the endomembrane system, cell cycle, proliferative state, etc. From these markers of the cellular state, we extract multiple features using image processing, which we extract at the multicellular scale, also at the single cell scale, as well as at the subcellular scale. And using a large number of these features, we generate what we call a cellular state space. So here you see a map of the cellular state space where each individual cell's location in this space reflects a certain combination of the features that we extract from these cells. Now, when you look at this cellular state space, you can then also appreciate that this really shaped specifically by different properties appearing at different spatial scales. So there are multicellular properties that are important in shaping this space, single cell properties, but also subcellular features that contribute to that. So once we were able to do that, we asked the following question. Taking a cellular state space and projecting on that the activity of signaling nodes in a signaling network activated by growth factor, epidermal growth factor, do we actually see a certain pattern emerging when we overlay these activities on this state space? So I need to emphasize we did not use these measurements to create the cellular state space. That was really used by cellular state markers. We just overlay these signals now here on top of this space. And what you can nicely see is that different signaling nodes in the network are very nicely shaped along this state space. So in other words, position in cellular state space really drives what is the level of activation of your signaling node. To the extent that we could actually build fairly simple data-driven modeling approaches based on multilinear regression that allowed us to predict with very high accuracy what will be the amount of phosphorylated ERK, for instance, in an individual cell, when knowing its position in cellular state space. And when you project these predicted features or the predicted values on top of the state space and compare it to the measured values, you see that it is very similar. And if you do a side-by-side projection between measured and predicted levels of phosphorylated ERK, there's almost you're almost not able to distinguish these two. So we get a very accurate prediction of signaling nodes. And even though the activity of signaling nodes is highly heterogeneous, we can basically show that this is really driven by the cellular state an individual cell is in. So that's a contextual way, right? It's a contextual information processing by these signaling nodes. The second concept I want to introduce quickly is called multimodal perception. Again, something very well studied in the cognitive sciences, which basically reflects the fact that we use multiple sensory inputs that together allow us to create sort of a comprehensive picture of the world around us. And one of the key elements of multimodal perception is that it is partially non-redundant. So there is some redundancy in this system, which gives robustness. It allows you to be insensitive to uncertainties in individual inputs, but the non-redundant element gives comprehensiveness. It allows you to integrate multiple sources of different information to get a more comprehensive picture. So we asked, do actually individual cells do that? And in order to do that, we basically took our data and we asked for individual signaling nodes, as you see here on the vertical axis, whether they are conditioned by specific features of the multicellular, cellular, or subcellular state. And when you look at this, you can see that there's some similarity between signaling nodes, but overall, signaling nodes are quite uniquely conditioned by specific features of the cellular state or multicellular state. So in other words, each signaling node in the network captures, to some extent, non-redundant information about the cellular state. So given this observation, we then asked, maybe collectively signaling nodes can then capture enough information about cellular state space to actually allow individual cells to quite accurately know their position in cellular state space. So to do that, what we asked, we took, for instance, an individual signaling node like phosphorylated levels of ERK and asked to which extent can individual cells know on the basis of those levels where it is sitting in cellular state space. And if you see the color red, it means it can map the local neighborhood very accurately. And when you see colors down to blue, it says that it cannot map the neighborhood very accurately. So if you allow cells to map their cellular state neighborhood based on phosphorylated levels of ERK, you see that it can map this region, but not so much the rest of cellular state space. If you take it from another signaling node, for instance, phosphorylated mTOR, you see that it can map different regions of cellular state space. Together, they actually synergize to already map quite a comprehensive picture of cellular state space. But if we take the information of all signaling nodes, we realize that it really maps quite accurately the full cellular state space. Now, this is very interesting, but of course the key question is, do single cells actually use this to make contextual decisions? So to analyze that, we sort of looked at it from this way. So what I just told you before is that based on position in state space, right? So let's say your cell in state 1 versus cell in state 2, you will adopt a certain position in signaling space, or in other words, your signaling nodes are conditioned to that specific state in specific ways. And depending on that position in signaling space, you will make a certain decision. For instance, re-entry into the cell cycle, which we can stain by having high levels of phosphorylated RB. Now, when you do this, when you overlay this decision for cells to re-enter the cell cycle, you see that as a function of growth factor concentration, you see that single cells decide to re-enter the cell cycle quite heterogeneously, but also shaped by position on cellular state space, as you see here. So we can actually very nicely predict which cells decide to re-enter the cell cycle based on its position in cellular state space, but also by knowing its multimodal percept, so knowing the activity levels of all the signaling nodes, as you can also see quantified here. So this idea that I show here on the left really seems to hold true, right? Cells make decisions based on their position in signaling space, which is in turn conditioned by positions in state space. Now, to test this functionally, we decided to use a perturbation approach. So in other words, can we have perturbations that allow us to not alter the cellular state, but to alter the position of these cells in signaling space? If we can do that, we can then, for an individual cell that has a certain state, we should then be able to push it to make a different decision. And indeed, we can show that this works quite nicely, because when we try to predict the decision of individual cells to become phospho-RB positive or negatives using models trained on cellular state space, we of course can do that nicely if we do not do any perturbation, but we start to fail to predict that when we inhibit signaling nodes. That is because we have moved cells in signaling space, which of course cannot be anticipated from models that were trained on unperturbed state space. However, when we use models that were trained on signaling space, and of course feeding now the new position of a cell that it has in signaling space, these models still remain quite a high accuracy in predicting this decision making. So what does this now mean? And this is my final data slide. What it means is that based on the position of cells in cellular state space, they will actually make quite different responses to drugs. And I would like to highlight two specific cellular state classes, basically class 2 that you see here. These cells will re-enter the cell cycle upon stimulation with EGF. That cannot be inhibited by an AKT inhibitor alone or a MEK inhibitor alone, but only by combining these inhibitors. And you can see that very nicely here. So even though these cells are in this location in state space and they stay there in all these conditions, you see that they really change their position in signaling space. So upon AKT inhibition, they end up here. MEK inhibition, they end up here, and only the combined inhibition brings them across this decision boundary, which is indicated by this dashed line. And finally, class 4, these are cells that are located here in state space. As you can see, these are actually typically phospho-RB negative upon stimulation with EGF, but interestingly, when we inhibit AKT, depending on this state, they will actually end up becoming phospho-RB positive. So this is of course bad news, because it means that we induce cells that are normally not cycling to re-enter the cell cycle upon inhibition of AKT. So to summarize what I showed you, by placing signaling responses in single cells within the context of their physicochemical state shows that heterogeneity contains additional information. I also showed that the concepts of contextual information processing and multimodal perception apply to molecular networks inside single cells, and that this reveals predictive rules by which properties at multiple scales determine single cell decision making and responses to drugs. Finally, I'd like to acknowledge my lab and the funding, and thank you for your attention.
Great. Thank you so much. Thank you so much, Lucas. We're going to hold questions, obviously, just like the other ones, until the conversation portion of things. And I see people are adding them to the board. So thank you for doing that. And I'll monitor that. So we'll go and dovetail right into our next speaker for this, Josh Leonard. Josh is at Northwestern University. He directs the biotechnology training program and is a founding member of Northwestern Center for Synthetic Biology. So that's exactly what he's going to be talking about today is synthetic biology and how we're using that for the tumor microenvironment. So Josh, we'll let you take it away. Thank you.
Great. Thank you, Julia. And thank you to Lucas and Michelle and all the organizers and everyone else for being here today. So I want to use my time today to just talk about a couple of technologies that I think could be useful for this problem that we've been talking about a bit today, but maybe even more interestingly for driving this conversation, I want to introduce two perspectives that apply to how synthetic biology may be able to apply to these things. So the first perspective that I'll start with is maybe the classic one. We're building tools, we're trying to make things that can solve therapeutic needs. And then I want to pivot to one that actually will resonate hopefully a lot with the talk that Lucas just beautifully introduced here, thinking about how that same toolbox can maybe be used as a unique way of studying natural systems. Okay, so briefly, I have a disclosure slide that some of the technologies I'll tell you about here have been commercialized here. And maybe the main science introduction slide here is a take on really that first classic perspective about synthetic biology. So, there are many ways of approaching these problems. I think the point I'd like to highlight is the text in red here is that in my lab, we work on gene and cell therapies writ large, but I think in general, we see our role here as accelerating the process between some kind of upstream insight from biology or medicine and building a thing that can test that idea as a therapeutic prototype or as really a conceptual manifestation. And how do you make that whole process faster? So we spend a lot of time building tools and conceptual and computational frameworks for making that prototyping process faster. And I'll mention two tools that I think have a lot of relevance for the problems we're talking about here today. So a big motivation for us is really the promise and the gaps in CAR T cell therapy, which Rogelio very beautifully introduced later today. And I'll just kind of highlight one that we've been spending a lot of time on, which is that for the special case or for the general case of solid tumors, just as a reminder, the barrier here is that you very seldomly have a unique antigen present on the tumor cells. It's shared at maybe different densities or just shared between healthy and tumor tissues. And so even if you have a nice CAR, it sometimes is inadequate for getting that safety to efficacy balance that's needed for getting therapeutic benefits. So how do we go about solving that? Well, we're going to talk about a slightly different approach for taking on a similar challenge here. And so just to kind of frame the way I'll be describing these tools, this is a very classic way that we think about how to build up a complicated functionality from parts here. And of course, this is easily described as a receptor signal transduction cascade and maybe gene regulation circuit, but it actually has power in the design framing to think about these in these kind of metaphors of sensing, processing, and actuation. And just to kind of illustrate a couple of approaches to that, I'll start with the first story and spend most of my time really at one that focuses on this information processing and actuation level. And then I'll close with just a brief mention of a technology that addresses the sensing area, which is complementary to some of the stuff we heard nicely presented by Xiaojing. So our theory that we wanted to try to implement here was that if you could make the activation of a CAR T cell therapy limited to the tumor microenvironment, then you could overcome the specificity challenge by making, for example, the CAR expressed only when it's in the presence of tumor microenvironment associated cues. And this is one beautiful story that kind of illustrated the potential of this approach by Carl Osti and by Kosti Arnold and company, showing that if you used a engineered hypoxia responsive promoter, you could limit the expression of that CAR to the tumor microenvironment. And that indeed gave you this nice selectivity for killing in the tumor environment versus in healthy cell environments that share that same antigen. And one of the things they noticed in this paper and kind of highlighted was that if you rely upon that native hypoxia response, this works in some cases and it doesn't work in other cases. And in particular, in cases where the tumors were modestly hypoxic, they didn't see efficacy in the same way that they saw with profoundly hypoxic tumors. And so we took this as one of those insights that we can now turn this into an engineering problem. So for example, what are the things we need to do to take that base hypoxia responsive promoter and change the performance characteristics? What are the things we can access? Maybe what are the things we can't access in order to make that technology more aligned with the performance needs of a therapeutic here. And so this story I'll tell you is one we published recently in ACS Synthetic Biology, focused on that goal. So in this case, here's a little preview of how hypoxia regulation works, or a summary of how hypoxia regulation works. An advantage of using this is it's a pretty well described system, pretty well understood. And the premise is that your cellular response to hypoxia has to be very rapid. So it's one of those responses where the proteins are synthesized all the time, but they are stabilized in the presence of hypoxia. That gives the ability of the cell to respond very quickly to the onset of hypoxia by essentially disrupting a basal degradation process here. And so our thought was, well, the basic mechanism is understood, at least many of the players are understood. Perhaps we can use genetic circuits as a way of exploring whether we can change the way these work, and in doing so, study a bit more the way hypoxia regulation works. So if you were to make a cartoon out of it, this is the way we might describe the native hypoxia system, where there are native factors regulating engineered promoters. But we can also build novel feedback motifs. And this is just a kind of cartoon of a few. You see, it gets kind of complicated kind of quickly, depending on how you choose which factors regulate the production of which, in this case, ectopic versions of those same native proteins. And so you're changing by doing this, both the dynamics and the topology of the control that is sort of prosthetically added on to the cell's native hypoxia regulation here. And so each one of these effectively probes, and by perturbing probes, the way natural regulation works. And the key questions are, can we use that to change output? Can we use that to build understanding? And so here's an example of what that looks like when you place these under two conditions here. One left graph is under hypoxia. The right graph is under physoxia. Sometimes people do this at 20% O2, but that's not really representative of the difference we're trying to strike here between healthy tissues and hypoxic tissues here. And the base case circuit is kind of an improved version of an engineered promoter. That's our left graph over here. And in the middle and the right, we have gene circuits now that have different types of feedback. I've selected two for presentation here. And you can see that what happens in both of the feedback conditions is we get both an acceleration of the output and an increase in the maximal level of gene expression when those cells are exposed to hypoxia. And looking over here at physoxia, you can notice these little blue curves, they do dip up a little bit over time. And we spent some time really figuring out how that works using local oxygen sensors on the plate to see what's happening. And what's really happening here is these cells are starting to grow in little clumps. And once they grow in a clump, you get a local hypoxia cue. So this is a real response. It's not an artifact. It just shows you how you can make these sensors very sensitive to, in this case, even local variation in the amount of hypoxia these cells are experiencing with an incubator that's otherwise set at a level that doesn't trigger them when they're at low density. So that's very interesting, and I think it has some utility. And of course, you can stop here and take this down the road of trying to see if you can use it for useful purposes. But we did a thing that I think is a really illustrative example of the power of trying to understand what's happening quantitatively and not just qualitatively. So we, over the years, have built a number of tools for making model development very rigorous and reproducible across different applications. And I just wanted to highlight kind of how that helped us understand what's going on here. So we went through a process that's described in the paper where you start with the simplest possible explanation and you only add complexity if it's absolutely necessary to explain the differences between your systems here. And we eventually came to a model that was capable of explaining the differences between our circuits here pretty well, I think, across these different applications. And in doing so, we were able to do a couple of really interesting things. There are a few sort of detailed debates in the literature as to how hypoxia works. These models were able to come down on the side of one of those explanations versus another. We also were able to postulate a novel mechanism. Basically, we had to postulate a novel mechanism about a key aspect about how the cell transitions from the acute phase hypoxia response to the chronic phase hypoxia response, which hasn't yet been explained mechanistically in the literature, but we can actually propose exactly where it must exist in order for us to strike the difference between these. And the point I wanted to make that really resonates with Luca's presentation here is if you think about these circuits as perturbations, we're getting the same sort of system-wide insights out that sometimes you have to spend a lot of time making many, many observations with many, many detailed metrics. But we didn't do anything like that in this case. We used perturbations to change the dynamics, but we measured a relatively small number of things, and that was still adequate to constrain the models in a way that I think is emblematic of how that type of perturbation can be a nice complement to other tools here. So I'll just wrap it up with a couple stories showing that it also helped us figure out what are the limits of that approach? And if we could make it better, what would we need? And so the last part of this story, we used a synthetic transcription factor toolkit we built a couple of years ago to try to make circuits that work even better. And so with that, you can make cartoons like this, where maybe you limit the use of those native hypoxia response elements to just the endogenous copies, and you build your circuits out of synthetic transcription factors to avoid some of the limitations we discovered in the first part of the story. And just as an example of what those circuits look like, here's a list of them, and talking through just kind of a couple of key bits. This red box on the left here highlights the performance of that best circuit from the first paper. And so on this scale, it's just looking pretty low, but it's actually the same performance as we saw last time. And the key thing is, by identifying and overcoming those barriers, you can actually access extremely improved levels of performance here. So in this case, this particular circuit gives us a much accelerated and much amplified level of feedback, even compared to some of those nice systems that we built in the last case, the open loop case shown in the last slide. So I'll just mention briefly that there are some other technologies that are out there, and I won't go into the details here, but there are some technologies that are useful for sensing things outside of the cell and for linking these sensing and information processing layers together. There's a veritable toolbox now of different ways of sensing this. And what we've worked on for a number of years is the specific challenge of sensing soluble factors. It turns out that the need for sensing soluble factors and transducing that into a synthetic gene circuit has been a fairly tricky aspect of this tool development process here. And so we've been working on this platform for a couple of years called the MESA architecture, which is designed to just be as independent of native signaling pathways as possible, in order to make it possible to put these parts together. And this series of references kind of just highlights that we've been able to make progress on each one of the pieces to bring down the background, to make the performance very good, and maybe most importantly, to make these things not finicky, so that they're not sensitive to the cell type you put them in, they're not sensitive to expression level, they just kind of work every time. And so the new version of that I just wanted to highlight to close this out is that we've applied those lessons to the conversion of native cytokine receptors into these synthetic receptors. And so you can get the benefit of nice signaling, especially I'd say of IL-10 and VEGF, which are of relevance for our conversation here. And you can introduce those sensing events into synthetic information processing circuits and so on. So I won't show all the data, those are available on bioRxiv, but to put a cartoon kind of claim here, you can do kind of conditional expression. So this is IL-10 induced expression of a CAR. You can put them together into circuits. You can put them into different cell types, including including T cells, as well as other kind of workhorse cells. And you can even change out the transcription factors to co-opt native regulation of choice and link those things together in new ways. And again, in this paper, we talked about this as being useful for applications, but I'd make the argument that these are also really just new ways to think about how you might perturb a system and even implement some of those interesting speculations that our previous conversation discussants were postulating, for thinking about if you were to change the aspects of the cancer progression or the immune response to cancer. These are maybe some parts that could be useful for implementing those and testing those hypotheses. So I'll stop here, thank my group for the work they did on these things, and look forward to talking about some of these in the discussion.
Perfect. Thank you both for your presentations. I think, I had prepared a few slides to kind of intertwine some of what we do in the lab, but because of time, and I want to make sure we cover the discussion board. So I do have slides that I'm happy to send to people if you want to reach out to me and get them to talk about some of how we use the spatial niches and how we think about spatial niches for TLS. The highlights that I'll say just to introduce the topic, and then I'll dovetail into the board, is that we really think a lot about spatial as cell-cell interactions, right, in the tumor microenvironment. So it's a bit different than the previous two talks. And so I think that's what brings the three of us together very nicely. And for a long time, without spatial imaging and really robust spatial imaging and spatial transcriptomics, we didn't know a lot about TLS in situ in patient samples. And so now in our group, we have designed TLS signatures directly via spatial transcriptomics. We've looked at those cellular interactions of those cells and understanding what's going on in them. We've been able to cross-apply spatial transcriptomic signatures to different solid tumors, and look at this at the single cell level and multiple solid tumors to really understand heterogeneity across these tertiary lymphoid structures. And finally, because we're interested in B cells, as mentioned in the beginning, we think a lot about what happens when a B cell gets into a structure or outside of a structure, and we can use spatial imaging and transcriptomics to really look at those molecular patterns and show what might be causing those differences based off of the niches that they're in. So we do something very different than the other two speakers, which is great. And this is why we're together in this collaborative session. So to kick it off, and with that in the background, I'd like to go to the board and just start with some of these questions that have kind of popped up. So I think I'll start with the opportunities, because I think there's two questions here from the opportunity standpoint that would be good for both of you to address. One is there an opportunity to employ this cellular and signaling space to begin to consider biologically meaningful length scales? So Lucas, that's for you.
Yeah. No, that's actually a very important question. I do believe so, but I think we're still in the early days, and we also need to involve some solid theory here, because basically at the end of the day, we're still, almost everything we describe in biology is at the molecular scale. But there's of course length scales that are longer typically, right? Going to multi-protein structures, organelles, cytoskeleton, single cells, tissue, communities of cells that have of course a lot of functional information and causal effects on the behavior. But we have no real rigorous formal framework for defining that, unfortunately. And so that's what we need to do. We need to quantify features at these different length scales. We need to quantify how much information do they carry. How can we connect that to, for instance, the molecular scales? But I think this type of approach can get us there. And then obviously that's more from the measurement side. I think the perturbation side is a whole other world, of course. And there, I guess, synthetic biology can also really help.
Yeah. Josh, did you have anything to add to that before we move forward?
Not on that one, I think. Thanks.
Okay. And I think the other thing, just to kind of dovetail off of that opportunity question and think a little bit about incorporating technologies together, right? So I think, if you think about both talks, is there a way to take what's happening with synthetic circuits and how that's changing depending on cell state, and then maybe to take that a step further, which is something I'm really interested in, and I got thinking about while Lucas was talking, and actually it's up here as well, so that's great, is about incorporating it to where things are spatially located, which is what we think about a lot in the tumor, right? Like we care about where the cells are, and particularly we think about the immune cells and their crosstalk with stromal cells. So the keynote was really, really timely for that, right? But like, what do you think about incorporating these different techniques, downstream? Would love to hear thoughts about that, because I think that's an overlying theme here today.
Yeah. Do you mind if I start, Lucas? Maybe with the
no, go ahead,
that one.
go ahead.
I was just thinking, and then I'll throw it over. So this issue of, first of all, the first part of your question, which was to what extent does cell state maybe impact the performance of the circuits? It's extremely important. So I didn't highlight that, but that's something that we have studied in detail in some other cases in kind of an interesting way. So the way that it seems to be most important is in terms of overall expression capability. So ribosome copy number, sort of like metabolic activity. There are variations in any population of cells. And one of the things that we learned by building these circuits a couple of different ways is that the best way of building a circuit that works well is not to hope for a good state, but rather build a circuit that works across multiple states. And so one of the interesting things we found by doing some of these very complicated logic circuits, just to push the limit of how far we could do this, is that you can actually get a circuit that will perform two different calculations depending on the overall expression level of the components. That was not, we don't know how to do that on purpose yet. We did that by accident. We were going through and doing some characterizations, but it's definitely something that cells do, right? You can access similar kind of outcomes as a function of those kind of state changes. And one of the other questions that's on here that really relate to this like spatial aspect is hugely interesting, which is a big thing we've been working on with these receptors is making them more sensitive. And the synthetic ones are now about the same as a native receptor. And you can think about the overall sensitivity more as the combination of the receptor and the downstream cascade, right? That's the way we look at receptor signaling, or kind of like amount of data going through the cell normally. So that's nice, we can do that. But one of the things that's really hard to answer about what you need in vivo is that we don't know what a cell sees in vivo, right? Like we don't actually know the levels of signals, or when you get down to the spatial scale that's relevant for that signaling event. So to this question that maybe Lucas posted on here, I'm actually really excited to think about if you could really tune the sensitivity of these receptors to discriminate between various in different concentrations, you could use that as a local probe just to kind of add to what we can measure by other methodologies about how those variations at the small length scales might impact the environment cells are seeing.
Yeah, no, absolutely. I fully agree. And, I didn't highlight this in my talk, but for instance, when we look at which cellular state features are really conditioning signaling responses, then if you look, for instance, at, the ERK pathway, it's highly conditioned by, for instance, the endosomal pathway. And the, membrane trafficking endocytosis is highly varying depending on where you're sitting in a tissue. When cells are highly crowded, their endocytic pathway is in a very different mode than when cells are spreading out and migrating, which of course directly impacts, which molecules will be meeting, which adapters will be binding to ERK, for instance. And so that's key, and that's complex, right? But you can start delineating that by doing this multi-scale image-based approach. And then, of course, if you synthetically can tune sort of the localization of for instance, ERK as an example, in a synthetic manner, you could actually manipulate that, right? That would be quite cool, actually.
Do you mind if I build on that? I had a question for you, Lucas, that kind of builds on this. Have you ever applied sort of an information theory framework to the way that might affect the amount of information that is being passed through these networks as a function of some of the layers you introduced in your analysis?
Yeah, absolutely. I mean, so it's not something, again, I mentioned in the talk, but in the paper, we actually applied an information theoretical framework to quantify sort of the information bandwidth, the number of bits, if you wish, that cells can process. And if you take a non-contextual view on this, you actually think that cells have a relatively poor information processing capacity, because there's lots of heterogeneity. And so if you look at different levels of growth factor, you see that these distributions overlap. However, if you place it within the context of the cellular state, you actually realize that heterogeneity doesn't mean uncertainty. It actually means additional information. So in other words, we are now starting to say, I mean, certainly much more work is needed, but the way we look at signaling networks is, yes, they capture information about growth factor, but also, and perhaps even primarily, about the physical chemical state of the cell. And so collectively, that is actually a very large of information that these networks can capture, right? So we're definitely interested in using such approaches, theoretical approaches. Yeah.
Cool. Thank you.
Yeah, and I think also thinking about the theoretical approaches, there's been a lot of comments on perturbation models and perturbing some of these different, aspects that you're talking about. So maybe you could share your thoughts on models that are out there. Like I know from the spatial standpoint, for what we think about, there's a lot of perturbation models where you can look at the perturbation of the tumor cell for specific axes, receptor ligand interactions, and then see by perturbing those tumor cells what happens as a result. And of course, this is more in the mouse model, right? Not in humans, because you have to put it into an animal. But just wondering your thoughts for your two fields, kind of, where you see that going, and if you think that might be an opportunity.
Yeah, I mean, well, with respect to perturbations, of course, there are many pharmacological ways of perturbing the cellular state. They are unfortunately also typically quite toxic, right? I mean, many chemotherapies, think about paclitaxel, is of course in the end perturbing the cytoskeleton, right? And of course you have many molecules that can perturb the endomembrane system, et cetera. But I think low doses of these drugs in combination with targeted therapies in cancer could be a very promising approach going forward, right? Because I think manipulating the cellular state is just as important as manipulating these signaling pathways.
Right.
Yeah.
Yeah, maybe I'll add on to that. I think when we started getting into this, we thought about perturbations on the systems biology side. We were inspired by some of these classical approaches where you use really well-developed networks of agonists and antagonists, and you can use multi-parameter flow cytometry to get many aspects of cell state. And we're thinking about what are the analogs as you scale up to be able to do those perturbations at a tissue or an animal scale. And one of the things we were excited about is this idea that if you want to make a perturbation in the way this multicellular network works, then you're kind of changing the input-output relationships of various different cells in the network. And so one of the early things we did with one of these engineered receptors, not the really great ones that we have now, are just come up with new input-output functions. Like if you have a theory that perhaps it matters that macrophages respond to an IL-10Q and produce a given cytokine, you can test that by having them produce 10 other different cytokines in different models and see to what extent the immune system we have and the immune system that could exist do different things phenomenologically. And so it's actually a really complicated thing to do. It's like a pretty expensive experiment to do that kind of a discovery process at the level of that kind of perturbation. But I think it's got a lot of promise if the motivation were there to do it. And I think the tools you heard across many talks today, I think we're, if not there, really close to there to be able to run an experiment like that.
Yeah, agreed. And we're coming up on time and we have to have a break so everybody can stretch before the collaborative session. But I did have one other thing and one other thought that I think encompasses some of the final things that are up on the board, which is, we often think about, certain aspects of the tumor microenvironment in a silo, right? And so if one person thinks about the tumor, one person thinks about the immune system, one person thinks about the stroma. And so I thought one of the questions that's on here is and this is more for cell state, but like, what are the inputs for cell state? And do they include certain things that would affect cellular state, such as the ECM and things like that? And I think, we can have a short answer to each of those, but I think it's really important, right, to look at that interplay. Like as somebody that was bred as a cancer immunologist, I don't think just about the immunology side of things. I don't just think about the B cell and the TLS. I think about how the stroma affects it. I think about how the tumor affects it. So maybe you can touch on how these can be like kind of multidisciplinary across cells, right? And thinking of that kind of cooperative nature or uncooperative nature within a tumor.
I mean, I fully agree that of course the tissue microenvironment will, and as has been shown already in many papers across many years, that has a huge effect, of course, on the mechanical state of cells, on the metabolic state of cells, etc. And so, well, the way I want to sort of look at it, and of course most of our experiments are done in very controlled conditions on tissue culture cells, right? So it doesn't have the same complexity as a tissue. But overall, one really needs to take a multi-scale and multivariate approach, right? I mean, biological systems are not univariate, they're not bivariate, they're really highly multivariate, and you need to capture that information. Unfortunately, maybe we still don't know exactly where is all the relevant information, but my hope is that at some point we get to that level, that we understand which information at which different scales we need to capture to really get to a comprehensive picture, right? And you need to have that, because if you do your studies in a non-contextual setting, you will really miss, there are hidden variables that you're not getting, and you can really make wrong interpretations, right? So, but that's more for the future. But I definitely say that integrating these spatial skills is going to be key. Yeah.
100%. Great. Well, fantastic. We are a little bit over, but we have now, I think, a break until 3:35, where we'll reconvene and have a wrap-up discussion. I do want to apologize that I will be darting out of that because I have to go pick up my children, but it's been a pleasure to hear everything and see all the great advances that are happening, and I really hope that it's fruitful. Thank you so much.
All right. Thank you. Bye.
Thank you, Julia, and thank you to the speakers as well. Again, we'll be reconvening at 3:35, as Julia mentioned. During that time, again, I just want to remind everyone to continue to add your ideas with respect to challenges and opportunities to the Miro board. See you also.
Just a quick note, Josh. I'm not sure if our breakout room did end.
Or if it goes on.
Oh, there you go. Thank you.
Do we just wait a few more seconds until everyone are back in?
I think they were all kicked back. So when you're ready, I think you'll have who you'll have.
All right, so thank you all for joining us for the final session of the day. This would be a discussion session, and I'm hoping in a few moments we'll have all the panel members highlighted in the in WebEx. So we are fortunate to have Dr. Thea Tistri, Josh Leonard, Jose Rees, Evanthea Russo Torres, and Ellen Pure with us. I just wanted to confirm if Dr. Ellen Pure is here. As we did not get a chance to formally introduce Dr. Pure earlier, I wanted to take a moment to do so now. Dr. Ellen Pure is a professor of biomedical sciences at the University of Pennsylvania. Her research focuses on the role of stromal cells and extracellular matrix and inflammation, fibrosis, and cancer. We are thrilled to have her today to share her perspective and join the discussion. Thank you so much for being here, Dr. Pure.
Thanks for having me. Sorry I wasn't here at the start. Their flights into Philadelphia are currently barely making it in. So
thank you so much. All right, so over the course of today's talks, we have heard an inspiring range of perspectives spanning from immunology, systems biology, and synthetic biology, and how these disciplines are being applied to understand cancer. And I would really like to use this time to focus our discussion on the opportunities and challenges involved in kind of integrating these fields to tackle, tumor immunology. Ellen, if I may start with you, and of course, I invite everyone else to jump in. How do from a broader perspective, how do you think we can begin to kind of systematically and more importantly, functionally map these kind of multicellular communications that we often see within the tumor microenvironment? And in your view, how can systems and synthetic biology contribute to this effort?
that's a very, very focused, small question now. No, I think one of the issues, and I just came back from another meeting where we're having similar discussions, of course, which is, what are the model systems that we're using, right? I think that's going to be a very important issue. And I think they're complementary in many ways, but, there are also issues with each of the situations, animal models, organoids. I think now, I also think going between species, how many things are not translating from efficacy in a mouse model to a human system. We all know that problem. And I think we really have to think about, and then reconstituting either through organoids or other systems, putting up the compartments back together. And then I also think right now we're mainly talking three compartments: the tumor cells, the immune cells, both innate and adaptive. We got those both covered, and we're talking about stroma and matrix. And I think we all know that now there's clearly very, very strong evidence for a fourth compartment that we're still not integrating with, which is innervation with nerve system, right? I think that's going to be the next thing that we're not modeling right now. And so I guess as hard as it is, it's important to not only use reductionist and reconstitution approaches, but also, especially in all models, but human, is getting intact tissue and leaving them intact, right? And then, so to do that with human, you lose things like, so you can use precision cut tissue slices, which I think is a useful model now, but of course you lose the blood flow. So you have to use complementary models, but I think that we need both the reductionists to test direct interactions and how they work. But I also think we have to get back to the more undisturbed architecture and physical properties, and try to make sure that they work there too and in human systems, right? Because I think otherwise we're spending many hundreds of millions, if not billions of dollars on things that don't end up translating in the patient. But I also think that, all this being so complex and having such complex networks, we do need these systematic approaches and computational approaches. And I think that, but right now we don't have sufficient data to put in to get the answers, the correct answers necessarily out, right? Because all of that depends on what the data is you put in for it to work with, right? So I think we have to really think about that a lot. But I also think we have to start thinking sooner rather than later that we've left out yet another just as important compartment. So we don't spend too much time ignoring it before we put it all back together. And how can we do that? I mean, I think those are important questions too. But I think mostly we need complementary data sets that are, like I said, from different model systems, and then the most robust ones are those that reproduce in multiple model systems, right? I think we have to really think about that.
Wonderful. I do think like there's a lot of like recent consortiums, especially trying to collect these large data sets at the tissue level. But again, those are not, we cannot photo them. I liked, Evanthea previously mentioned, using these kind of systems biology approaches, we could potentially narrow down some of the targets using these kind of observational studies. Josh, do you think like how, even when we narrow down, it's still a lot of, different things that we want to look at, right? So we can probably go from like, 20,000 observations to like 100 observations or 100 different hypotheses that we want to test. Do current kind of synthetic biology, approaches allow us to, multiplex these kind of tests, or do we have to go with like one test at a time? Like, do we have to engineer one receptor, one ligand, test it, and then move to the next one, or can we multiplex them?
It's a great idea. Were you directing that to me?
Yes.
Yeah. So we actually came up with that question or idea at our little breakout session too. I think the idea is probably yes. I think it's doable, right? I mean, a great example of kind of where that's been done is the pooled CAR T cell screens, which have been done in vivo to just examine whether or not you can separate phenotype at the level of the mouse model anyway. So I think that's an example of a yes. The combinatorial perturbation topic that we came up in our breakout, I've not seen that done, but certainly is compatible with methods that exist, at least in some places. I think the challenge is genetic screens are easier because you have the same tool. It's just a guide RNA or whatnot that goes into every different variant. I think that CAR example was one where you have to synthesize a novel variant for each one of your screen members. So probably the bottleneck is the availability of the parts library that maybe answers your question, but then the implementation is probably mappable onto those other methodologies.
So can I ask all you that do the systems biology, my view of it is that we need an iterative approach, right? So you get some ideas of pathways that are important. We then go perturb them biologically, and then we give the answer back, right? Doesn't that then refine? So I guess for me, if I understand what you all do, is that that's what we need, is much more of this iterative process that, you give us these pathways that look prevalent, and then we go and test them, and then it needs to be fed right back into that to see what held up, what needs to be adjusted, and so on. And I'd love to have more of a view of how that could work more efficiently between those doing those biological questions and back in an iterative process.
I guess from a perspective from someone who's using the system, using the brilliance of the systems biologists, I think you're right. It is iterative because we still have to, the input still has to be, still has to come from a lot of the wet lab work that we do. But where I see it being, like where I see the efficiency, really is when we can run simulations to give us answers to the biological or immunologic questions that come from what we've learned mechanistically. So if we input, let's say we figure out one piece of the mechanism, like we figure out a specific receptor that when blocked changes or has a really significant effect on suppression. If we account for that in an iterative version of our math model, but then what we use it for is to run simulations, like in our breakout session, like we were discussing, run simulations on how that affects tumor cell growth in different metastatic sites, which would take a really long time to do, or be tons of resources. So the use of the systems biology in this case is actually on the output of what the math model can do. But in order to really make it biologically relevant, you still have to have that iterative approach to specifically inform the parameters in the model.
And I'm kind of curious, maybe from the NCI side, this seems like it's really a matter of getting people together and getting communities together as well. Are there I don't know, ideas or things that have worked well in the past, maybe from other disciplines, to get more of this back and forth?
Yeah, I guess the other thing that came up in this other thing that's really struck me, which I think is true, is in that iterative process, just as much as getting validation data that a pathway did do what we might have predicted or changed things differently, and then getting that back into the, equation, is the things that don't work. I think that's another really big issue. We don't tend to report things that don't validate biologically. And, that is just as important, right? If it doesn't work and we're off, we've got to accommodate that in what we're looking at. I don't know the best way to do that because it's hard to publish these things that are negative results. More and more it is happening, but I just think that there has to be a way that there are consortium, like you say, that stay in touch enough where both that validation gets reworked in an iterative process, but also where it doesn't work. I think that's so key.
Yeah, Ellen, can I build on? I wanted to ask this as a follow-on to something you said at the beginning here, and maybe this is for everybody. I'm curious about this. We have all these debates in the translational space around which is the best model for going to evaluate one of these therapies in a pre-human model, and people fight about whether this mouse model or that mouse model or non-human primate or whatnot. And the answer is like none of them actually predict for a lot of therapies, to the point where people are burned out doing anything between in vitro and people. And I just am wondering, do we have knowledge as to like how much effort do we put into this kind of idea of figuring out why a model fails, to understand whether an in vitro model at organoid at better mouse is a solution, or just if it isn't, how do we make the clinical evaluation plan somehow better aligned with the technical risk associated with those different products? Do you know what I'm saying? Like, I don't know how much we know about why things fail.
No, I agree, but I think that it is important to figure out why some things don't work, right? But like I said, I would always complement it with trying to use some human tissue as well. Again, the big problem there is you're never going to know for sure till you go into a human. It's just till you do a clinical trial, we're never going to know for sure. But I think if we can build enough through the systems biology and enough of these scenarios you can run in computational biology, is it use some human tissues, knowing that you lose certain aspects. And like I said, using data from multiple models, ones where you don't lose flow, trying to do tumors on a chip where you can add back flow in one of the reconstitution models, using multiple models and going for those and adding, but we're never, in my opinion, I think until we can completely, the variation from individual to individual and that life history of their immune system and their microbiota and timing and everything, I don't think we're, at least not in my lifetime, are we going to be able to know for sure till we go into a patient, quite frankly. And the other thing I've been worrying about a lot lately is, when we go into human on a clinical trial, most often they have failed multiple lines of therapy, especially in a tumor, and the immune response to the tumor, as well as the stroma and the innervation and so on, is going to be very much more influenced than we yet understand by those prior therapies, right? And in aged people, as opposed to a lot of the studies which are done in younger mice, which is an issue. That's an easy one to fix. You go do aged mice. But this idea that we go into naive animals and treat tumors that haven't been, gone through all these machinations, which clearly will kill off some tumor cells, causing differences in the immune response and so on. And I don't think we've nearly taken into account enough the idea that some of these failures may be because of that. We haven't really thought about that much, right? at least as I'm aware.
I see
Yeah.
Lucas Pelkmans has his hand up. Go ahead.
I mean, and maybe I would like to add something here. I mean, at least here in Switzerland, what we're trying to do more and more is observational clinical trials where we actually get biopsies from patients. Of course, this typically works with tumors that are sizable, so we can get enough material. With ovarian cancer, melanoma, it works quite well. And we really can ex vivo do a lot of things now with these cells or with these tissue samples. In an iterative manner, right? So we can systematically screen, a couple of hundred drugs. We can really look at how do these cells respond to these drugs. And I think these ex vivo models are very informative, and also they reveal so much heterogeneity between patients that we are not yet even fully grasping. We're understanding heterogeneity within tumors, single cell heterogeneity, microenvironment, et cetera, but then there is this interpatient heterogeneity, which is also huge. Which we all need to map still, I think.
I agree.
Great point.
I would also just add to your point, Ellen, about the negative results. I think that's what's actually so attractive to to me as someone, as a tumor immunologist trying to, using systems biology, is that trying to actually figure out why something didn't work, to suggest something that, a reason for either you could spin it as a mechanism of resistance, although you'd have to do a lot to prove that, or at least to suggest in a discussion, let's say, what could be evaluated next. When you put it in the context of modeling, it actually turns it into a positive result of some kind. Well, the result was negative. The survival study didn't work. The mechanistic, the inhibition or knockdown of that receptor didn't do anything. We put it into this math model, and in fact, we found out that that's right. You couldn't just do that one thing to change survival, but actually, if we change this receptor and this receptor and this receptor, all three of them, it did work better, or at least the model predicts it will. So you have at least something to say that's not fully negative data. And I think in that regard, it's a very attractive collaboration between fields, because that's again a way to kind of be more efficient in our reporting, so that we don't have to leave out the negative data just because it's negative data. We can put it in and say, in fact, yeah, we didn't identify the one mechanism of action of this drug. What we found out was you need this drug to do all four of these things to see a result on tumor cell growth. And we didn't do that here in this paper yet, but at least we have an idea of what we would do next.
Yeah, I think that's really key also, because I think that everything we know says that we're going to need to use combinatorial therapies to really be successful in treating cancer, right? Whether they're combinatorial simultaneously and or sequentially, that's really key. And I think maybe that's where the modeling can help a lot, right?
Yeah.
The systems modeling.
Totally.
Yeah, can we do 10 different treatment categories, just varying either the order in which we give a drug or how long we give a drug for before we give the next one, where, if you were to try and do that actually in a wet lab setting, it'd be, again, just really onerous, but you could run those simulations and 10 minutes later you'd have results. And
That's where I thought Lucas's talk.
Yeah. No, I mean, one can run quite a bit in the lab these days.
That's where I thought
right?
Lucas's study was very.
Go ahead, Lucas. You were saying?
Yeah, sorry. No, I mean, of course, one can, with modeling, explore a much vaster space of potential, conditions. But there's sort of this medium scale throughput we can now really do with multiplexing capabilities that really generate lots of data. And that's not just sequencing data, right? That's really phenotypic responses, molecular responses, multiple nodes and networks. So it gives a lot of rich data for actually feeding those models, right? Also, absolutely training with AI these models. I'm sure we can do that now. Definitely a great opportunity.
In the final remaining minutes, I kind of want to change topics. maybe a bit of an off-the-wall question, but open to everyone. One thing the workshop is spatiotemporal processes in cancer. We haven't talked too much about the temporal, and I think it's very interesting. And a little bit, Dr. Chilsey's talk really highlighted the central importance of chronic versus transient inflammation. Time is something that is, I think, understudied, undercontrolled. We can obviously control things temporally with things like optogenetics or related technologies, but you can also build circuits that respond to time. So you could also imagine you could build a cell that responds to chronic inflammation, but not transient, things like this. So I guess with kind of blue sky, anything is possible type of thinking, what would be the most interesting way to use time to maybe, understand or address cancer? Thinking about tumor-immune interactions.
Something that I've been thinking is how does both during tumor initiation, for example, how does mutations affect the probability of a cell becoming cancer depending on the cell state that it is? And of course, these cell state transitions are evolving over time, but you can also think about it in terms of therapy. So for example, what Ellen mentioned about the idea of implementing sequential therapies that may depend on the state that the cell is in, I think is really important. And of course, characterizing the dynamics of cell state transitions will be really key to sort of tackle these challenges.
Yeah, truly, I do think just measuring how long a cell can stay in a particular state before it switches would also be very important. All right. Should we are almost right on time with 1 minute left. I'll probably take that time to thank all our panelists for such a thoughtful and kind of engaging discussion, and thank you for staying all through the day, and to all the speakers and NCI staff and everyone who joined us today. It's been incredibly valuable to hear these kind of very interdisciplinary perspectives on tumor-immune interactions and opportunities at the interface of spatial systems and synthetic biology. Again, as a reminder, our final workshop in the series will be held on May 20. We'll be focusing on therapeutic resistance at that point in time. We hope to see you all back there. Thank you again for joining us. See you all soon.
Thank you.
Thank you.
Thank you.
Thank you all.
Bye.
Bye.